In the deep water, far below the last blue memory of sunlight, a body begins to fall. It is enormous, longer than 3 school buses laid end to end, wider at the shoulder than a doorway is tall, weighing more than 100 tons while it was alive and breathing at the surface. Now it hangs in the water column, suspended between what it was and what it is about to become, and the dark around it is complete and cold and absolute. There is no light here. There has been no light here at this depth since the ocean formed. The pressure against every surface of the body is immense and even and still, pressing from all sides with the calm indifference of a mile of water overhead, and the body slowly begins to sink. For days before this moment, the body floated, the great engine of decomposition, which begins the instant a living thing stops living, had been filling the body from within, the bacteria already present in the gut and tissues multiplying rapidly, consuming soft material, releasing gases that inflated the carcass from the inside, the way a slow breath fills a sealed room. The body drifted at the surface, enormous and pale against the dark water below it, and for a time the ocean held it up. Seabirds descended, sharks circled. The exposed skin and blubber were opened by small acts of feeding, and salt water entered, and the gases began to dissipate, and the long, slow drinking of the deep ocean into the carcass began. The body grew heavier, not dramatically, not all at once, but in the patient arithmetic of density, water replacing gas, mass accumulating, the bones growing heavier than the water they displaced. And then the moment arrived, gradual and unannounced, when the body was no longer light enough to remain at the surface. It sinks at the rate of a slow elevator descending through a very tall building. 1 meter per second is too fast. 1 meter per minute is about right, unhurried, continuous, carrying all that mass downward through water that darkens by degrees as the surface recedes. The blue of the upper ocean fades to indigo, and then to a color for which there is no name, because no eye has ever seen it, and then to nothing at all. At 200 feet, the last violet light is gone. At 1,000 feet, the water is as dark as a closed room with no windows. But the body keeps going, pressing downward through the cold, through the dark, through the increasing pressure that does not stop building the further down it goes. The descent takes time. A body dying in water 2,000 meters deep may reach the seafloor in 20 minutes. It may take 40. The arithmetic of depth and speed means the journey is neither brief nor long. It is simply the time required for that much mass to travel, that much distance through cold water, steadily, silently, without anything to mark its progress, except the changing pressure and the slowly dropping temperature of the water through which it moves. There are no landmarks down here. There is no way to measure distance except by time, and time in this darkness has no natural unit. The body falls. The water holds it gently, resisting, pressing back from all sides, and still the body falls, and the surface grows farther away, and the bottom grows closer, though neither can be seen. Midwater is its own world, strange and sparsely inhabited, crossed by creatures that have evolved specifically for the featureless immensity between surface and floor. Some of them carry their own light in small cold lanterns mounted along their flanks, or suspended from modified fins in front of their mouths. Others move in complete darkness without any light at all, navigating by pressure waves and dissolved chemistry and the faint electrical fields generated by living bodies nearby. None of them are large. The water here is cold and very poor in nutrients, and the animals that live in it have grown small and slow metabolizing and patient, calibrated to a world where food arrives rarely and in small quantities. drifting downward from the productive surface in particles so fine that they are called marine snow. The accumulated dead cells and shed skins and fecal matter of the organisms living far above, falling for weeks before they reach this depth. The midwater creatures that live near the body's descending path may sense it as it passes. The chemical signals it releases, lipids dissolving into the water, proteins breaking down at the edges, diffuse outward in a faint plume that spreads horizontally in the slow currents. But nothing here is large enough or equipped to feed on what is passing. The body moves through their world and is gone before most of them have fully registered its presence. Below the mid-water zone, the cold intensifies. The temperature drops steadily as the depth increases, and by the time the body has fallen through the first 1,000 meters, it is moving through water that hovers just above freezing, water that has not seen the sun for centuries, because the circulation of the deep ocean moves so slowly that the water at abyssal depths is ancient water, water that sank at the poles under conditions that existed long before anyone alive today was born. This cold is not hostile, exactly. It is simply the cold of deep time, of water that has been turning slowly through the ocean's great conveyor belts for hundreds of years, and it has no interest in the body falling through it. The cold is just a fact. It will slow the decomposition of what it receives, and the slowness will matter enormously for what comes next. But that is not yet. That belongs to the floor, which is still coming. The sea floor in the deep ocean, in the region scientists call the bathyal zone, the zone between roughly 1,000 and 4,000 meters, where the most elaborate whale fall communities form, is a place of unusual stillness. The sediment there is fine and pale, the accumulated remains of microscopic organisms that lived and died in the sunlit surface waters, above and sank for weeks and months until they came to rest here, building up in layers so gradual that a single centimeter of sediment may represent centuries of accumulation. The floor is nearly flat in most places, gently rolling in others, dusted with this pale sediment that records the slow passage of time in its layers the way rings in wood record the years of a growing tree. There is no light. There is no sound except the slow movement of bottom currents, which in many parts of the deep ocean are almost imperceptibly slow. The pressure down here at 2,000 meters is roughly 200 times the pressure at the surface, enough to collapse a human lung to a small dense package in the time it takes to notice. But the animals that live here have spent their entire evolutionary history under this pressure, and to them it is simply the weight of the world. Very little food reaches the seafloor. This is perhaps the most important fact about the deep sea floor to understand before anything else. Not its darkness, not its pressure, not its cold, but its hunger. The marine snow that falls from above is sparse. By the time particles have sunk through 2,000 meters of water, most of the organic material they contained has been eaten, respired, and recycled by the midwater organisms through whose zones they fell. What arrives at the floor is a thin, dilute, continuous drizzle of organic matter, enough to sustain the sparse communities of deposit-feeding worms and sea cucumbers and bacterial mats that make up most of the deep sea floor's resident biology, but only barely. The organisms living here are calibrated to scarcity. They are slow. They are small, most of them. They wait. They process sediment for the faint traces of organic carbon embedded in it, extracting energy from material so dilute that it would be invisible to any instrument a surface ecologist might routinely carry. The deep sea floor is alive, genuinely and everywhere alive, but it is alive in the way a desert is alive, with organisms that have made precise and elegant accommodations to almost nothing. and then a whale arrives. The body, which has been falling for somewhere between 20 and 40 minutes, approaches the floor now. The bottom currents carry a faint chemical signal of what is coming, lipids dissolving, proteins fragmenting at the outer edges of the descending carcass, and perhaps some of the organisms in the surrounding sediment sense it before it arrives, though most of them are too small or too slow to move toward a signal, even if they detect one. The first sign is the pressure wave of something enormous displacing water. Then the shadow, if shadow means anything where there is no light to cast one. Then the impact, which is not violent. The water slows the descent in the final meters, and the body settles rather than crashes, pressing into the soft sediment in a slow billow of pale particulate matter that rises and spreads outward in a cloud, and then slowly, over hours, settles back down. The sediment records the impression. The weight of the body presses into the floor, and a stillness follows, brief and absolute, before the chemistry of the place begins to change. The carcass that has settled here is a fin whale, perhaps, or a blue whale, the largest animals that have ever lived on this planet. If it is a blue whale at the larger end of the species size range, it is 30 meters from rostrum to fluke, and its mass, even in death, is staggering. Tens of thousands of kilograms of soft tissue, muscle, organ, blubber, connective tissue still surround a skeleton whose individual bones are the size of furniture, of small cars, of things that have no good comparison in ordinary domestic experience. The skull alone would fill a modest room. The vertebrae, stacked along the central axis of the body, are each as wide as a kitchen table and several times as thick, their surfaces dense and hard, but their interiors, the cancellous matrix, the honeycomb of interconnected channels that make bone both light and strong, saturated with something that will fuel this place for decades. The interior of a whale's bone is not empty. It is not merely structural, not simply calcium and collagen arranged for strength. Deep inside the latticed matrix of a large baleen whale's vertebra, filling every small channel and cavity in the porous interior, is a pale wax-like material, lipid, oil, stored energy accumulated over years of feeding the krill and small fish and copepods of the productive surface ocean, converted by the whale's metabolism into fat and deposited in the bones as a long-term reserve. The bones of a large blue whale may contain between 2,000 and 3,000 kilograms of this stored lipid. The number deserves a moment of stillness. It is not a large number by some standards, but as a reservoir of chemical energy on the deep sea floor, where the annual input of organic carbon from marine snow might be measured in grams per square meter, several thousand kilograms of concentrated lipid represents something without parallel in this environment. It represents more carbon than the surrounding seafloor might receive in centuries of marine snow accumulation, delivered in one arrival, concentrated in one location, available to whatever organisms can find a way to use it. The bones hold this energy quietly. The skeleton of the settled whale does not announce what it contains. From the outside, seen in the imagination through the ROV cameras that would one day, not yet, this is the beginning, illuminate these places, the bones look like bones, dense, pale, where the dark sediment meets the lighter calcified surface, architecturally remarkable in their scale, but not visually different from what anyone might expect bone to look like. The oil is inside, the energy is stored. The decades-long slow release has not begun. The body is intact, the tissue still covering the skeleton, the blubber still thick around the flanks, and for now the place is simply a very large dead animal resting on the cold dark floor of the deep ocean, and the stillness around it is the stillness that precedes everything. It is worth staying with this stillness for a moment. The seafloor around the settled carcass has not yet changed. The deposit-feeding worms in the immediate vicinity of the impact are buried in their sediment, processing material from centuries before this whale was born. The bacterial mats that colonize any stable organic input have not yet formed, because there has not been time for even the fastest reproducing organisms to multiply to visible density. The chemical plume diffusing outward from the carcass is just beginning, a dissolved signal spreading in the slow bottom currents at a pace that will require hours or days to travel meaningful distances. Nothing yet knows, in any biological sense, what has arrived. The carcass rests on the floor, in complete darkness, under enormous pressure, in water just above freezing, and the future that is encoded in its lipid-saturated bones is entirely latent, waiting in the way that any stored energy waits, not wanting anything, not anticipating anything, simply present and available, with everything still ahead. The idea of a whale fall as a formal scientific concept is relatively recent. The recognition that a sunken whale carcass functions as a discrete temporary ecosystem capable of supporting hundreds of species through a succession of ecological stages spanning decades came into scientific focus in the late 1980s when the first systematic observations of deep-sea whale falls were made, and the full complexity of what was found there began to be understood. Before that, the fragments of evidence existed, bones recovered from the deep sea with unusual fauna attached, organisms found at vent sites whose presence at non-vent locations was unexplained, but the picture had not yet assembled itself into a coherent model. The model, when it came, was one of those clarifying ideas that makes a great many previously puzzling observations suddenly legible. The bones found on the deep sea floor, with their unusual passengers, were not anomalies. They were stages. They were moments in a long ecological sequence that had been running on the seafloor for as long as whales had been dying and sinking, long before anyone was watching. The bathyal zone between 1,000 and 4,000 meters is where the most ecologically elaborate of these communities form. The reasons are several, and they work together. The cold at these depths slows decomposition, allowing the ecosystem enough time to fully develop through its various stages. If the temperature were warmer and bacteria worked faster, the lipid reserves might be exhausted before the later stages could establish themselves. The pressure at these depths keeps the carcass from floating back up, which can happen in shallower water, where gas production during decomposition can reinflate the body and lift it toward the surface before the seafloor community is finished with it. The low oxygen in the sediment at these depths, the sediment immediately below and around the whale bones, becomes anoxic quickly as microbial activity consumes available oxygen, creates precisely the conditions needed for the anaerobic chemistry that will later power the ecosystem's most productive stage. And the distance from the surface means that the background input of organic material is at its most dilute, so the arrival of a whale is not just large, it is enormous relative to everything else available. A whale fall in shallower coastal water would be significant. A whale fall in the bathyal abyss is an event without local comparison. Slow. Everything about this process is slow. The sinking itself, at 1 to 2 meters per minute through 2,000 meters of water, takes the better part of an hour. The initial decomposition that bloated the carcass at the surface took days. The chemical plume, now spreading outward from the settled body, moves at the pace of deep ocean bottom currents, which in most parts of the abyss are barely perceptible, a few kilometers per day at best, less in calm periods. The sediment around the body will not change visibly for hours. The first large organisms to arrive, following chemical gradients through the darkness from kilometers away, will not arrive for hours more after that. And the longest phase of the ecosystem that will eventually occupy this place, the slow burning of lipids stored in the bones, the decades-long chemosynthetic community, will not begin until the soft tissue is entirely gone, which is a process that is itself measured in months. Nothing about a whale fall is fast, except by the geological standards of the deep sea floor, where everything is very slow indeed. A single large blue whale falling to the seafloor delivers, in one event, more concentrated organic carbon than the surrounding abyssal plain might accumulate from marine snow over centuries of quiet rain from above. That comparison is worth letting rest in the mind for a moment, as the carcass rests on the sediment. The deep sea floor around the body has been receiving its thin, steady drizzle of organic particles since long before this whale was born. The individual particles that make up that rain, a dead diatom, a fragment of copod exoskeleton, a fecal pellet from a midwater fish, each carry a tiny, almost vanishingly small load of organic carbon. They fall and settle and are processed by bacteria and worms and sea cucumbers in a community calibrated precisely to that level of input. And then the whale arrives, and in the instant of its settling, the energy budget of that small patch of seafloor is transformed by a factor that has no good analogy in the experience of the organisms that live there. They have been receiving background noise for centuries. Now something enormous has landed. The bones will be the last part of this to yield, long after the soft tissue is gone, long after the blubber and muscle and organ have been taken by the first wave of scavengers and colonists. Becaned, the bones will still be releasing their stored lipids slowly into the surrounding sediment and water, feeding a community of microbial specialists and the animals that depend on them, sustaining an ecosystem that could not exist anywhere else within many kilometers of ocean floor. The bones are not the structural remnant of the ecosystem, they are its fuel. The skeleton that lies at the center of everything is a slow-burning chemical engine, and it will run for decades, for 50 years, possibly longer, before it is fully spent. All of that future is present right now in the pale wax stored in the cancellous matrix of every vertebra, every rib, every dense slab of skull bone pressed into the sediment of the deep sea floor. The whale is at rest. The sediment has settled back around the impression of its body, the fine pale particles that were disturbed by its arrival, drifting back down over the course of an hour, covering the immediate area in a thin new layer. The bottom currents that have been flowing slowly across this patch of seafloor for centuries continue to flow, carrying now a faint chemical trace of something extraordinary outward into the darkness in every direction. The cold presses evenly from all sides. The pressure does not change. There is no light, and there has been no light here since the ocean filled this basin, and there will be no light here for as long as the ocean remains. Nothing moves except the slow current and the slow drift of particles and the slow diffusion of dissolved chemistry through very cold, very still water. The whale lies on the floor of a world that was here long before it was born, and that will be here long after the last trace of it has been incorporated into the bodies of things that were not yet alive. Its bones, those hollow lipid-filled chambers that kept it buoyant and flexible throughout decades of diving and surfacing, are already performing their next function, not swimming, not breathing, not moving through sunlit water in pursuit of krill, but simply being present and rich and available to the darkness. What lives in those bones is not yet alive here. What will grow across the sediment around the body has not yet begun to grow. What will arrive from kilometers away, following the dissolved signal, now spreading outward through the current, has not yet turned and begun moving in this direction. The ecosystem that this place is about to become exists right now, only in potential, encoded in the chemistry of stored lipid and the biology of organisms that do not yet know this place exists. The floor of the deep ocean is mostly barren. It stretches in every direction, flat and pale, and very nearly featureless, and its barrenness is not temporary and not the result of any recent disturbance. It is simply what the deep sea floor is in most places and most times, a vast cold desert receiving a thin, slow supply of organic matter from a world 2 kilometers above, sustaining life at a level of density and productivity that is almost incomprehensibly low by surface standards. Scattered across this desert at intervals that were once far more regular and are now far more sparse are places like this one, places where something large has arrived and transformed the local economy of the seafloor from scarcity to temporary abundance, from barren to rich, from the deep oceanic equivalent of nothing to something that supports hundreds of species and persists for decades. This is one of those places now. The whale arrived in darkness, without ceremony, settling into the soft sediment in a billow of pale particles, and came to rest. And it is resting still in the cold and the dark and the complete silence of the deep sea, with the future held inside it, the way that all futures are held, not separately from the present, not waiting in some other place, but right here in the chemical richness of a skeleton lying still on the floor of the ocean. The first creatures are already turning, somewhere in the darkness, following the slow chemical trace of dissolved lipid and amino acid spreading outward through the current. Something has sensed the signal. The message is faint and diffuse, attenuated by distance and diluted by the vastness of the water through which it moves. But it is there, and it is real, and it carries information that can be read by the right organism in the right way. And what that information says, in the oldest and most fundamental language of biology, is simply, here. Something is here. Something large and rich and full of energy has come to rest in the darkness, and here is where it is. But that arrival and everything that follows from it belongs to the long unfolding ahead. Right now, there is only the whale and the stillness and the slow breathing of the deep ocean around it. The lipid inside the bones is cold and still. The sediment lies undisturbed around the great curve of the ribs, the vertebrae, each one the size of something that would be remarkable to encounter anywhere on the surface of the world. Press quietly into the floor, their porous interiors saturated with oil that has been stored since the whale was feeding in sunlit water years and years ago. Oil that contains, in its molecular structure, the converted energy of krill and small fish from waters that were warm and bright and full of life, carried down here to this cold, dark place, held in bone, waiting without any awareness of waiting for the slow chemistry of the abyss to find it and begin. The whale is at rest on the sea floor. The darkness around it is complete. The cold is steady and even and deep. And inside the architecture of those enormous bones, in every small interconnected channel of the cancellous matrix, the pale stored oil is patient and still, holding within it the energy of a life that ended at the surface, and a future that will unfold here on the floor, slowly and completely, and over a span of time that is almost as long as a human life, a whole ecosystem, latent and waiting in the dark. From the settled carcass, something moves outward that no eye can follow. It is not light, it is not sound, it is a slow, invisible diffusion of dissolved molecules, fragments of amino acids, traces of lipids released as the body's outer layers begin their first quiet dissolution, spreading through cold water in every direction at once, thinning as they go, carried on faint currents that no instrument aboard any surface ship would detect. The plume has no sharp edge, it has no color. Where the whale's body meets the water, the boundary is already becoming uncertain, and the signal that carries farthest is the one that travels most slowly, molecule by molecule, through 3 kilometers of lightless sea. The seafloor around the carcass is still. The sediment that billowed when the body arrived has long since resettled. The fine gray dust has smoothed again across the bones of whale ribs and jaw, across the great curved architecture of the spine, across the wide flat plates of the skull, now pressed gently into the soft seafloor. Everything is dark, everything is cold, and into that darkness the plume spreads outward, a chemical message written in the only language the deep sea has ever used, the language of dissolved molecules in slow water, read by whatever has evolved to read it. What happens next unfolds not in minutes, but across time, in overlapping waves of life arriving and departing, each one different from the last, each one made possible only because of what came before. It is a succession. The word comes from ecology, where it names the orderly replacement of one biological community by another across time, and nowhere in the deep ocean does succession play out with more patience, more complexity, or more consequence than here at the site of a whale fall. The communities that will occupy this site will not all resemble one another. They will not want the same things. They will not be present at the same time. Each will do its work, and in doing it will change the conditions that allow the next to follow. There are 4 of these stages, 4 distinct ecological worlds, each one arising from the ruins of the last, each one lasting longer than a human lifetime in some cases, each one harboring species found nowhere else on the surrounding seafloor. The succession begins within hours of the carcass coming to rest. It does not end for decades. The first signal reaches something large before the first day is done. In the water column above and around the settled body, organisms capable of detecting dissolved organic compounds at extraordinary dilution have already begun to move. The plume has spread far enough. A current has carried some fraction of it toward a creature resting on the bottom, several kilometers distant. Resting is perhaps too gentle a word for what it does, because it is not truly at rest, its body making slow, continuous adjustments against the cold current, its sensory systems monitoring the water around it for exactly this kind of signal. When the dissolved molecules arrive, the response is not dramatic. There is no sudden acceleration, no visible urgency. There is simply a slow reorientation, and then a steady movement in the direction the concentration increases. This is the beginning of the mobile scavenger stage, the first of the 4 phases, and it is defined by motion, by the arrival of large motile organisms from the surrounding seafloor and water column, organisms capable of traveling kilometers in response to a chemical signal, capable of processing enormous quantities of soft tissue in a very short time, capable of reducing what was once the largest animal body in the ocean to a clean, pale skeleton in a matter of months. What the mobile scavengers do is violent in its aggregate effect, though none of them is violent individually. They feed, they consume, they return to the darkness between meals and come back and feed again. And each time they take soft tissue from the carcass, they open more surface area to the water, and the chemical signal strengthens, and more of them come. The mobile scavenger stage is the fastest of the 4 phases. It is the loudest, in ecological terms, the most visibly active, the most readily observed by the cameras of remotely operated vehicles that have documented whale falls in the Pacific and Atlantic. It operates at a scale that is hard to hold in mind. A large whale carcass may carry 40 or 50,000 kilograms of soft tissue, muscle, blubber, organ, and the scavenger community can remove the majority of that tissue within months, sometimes within weeks, under conditions of high scavenger density. The rate of removal is not slow. It is not gradual in the way the later stages are gradual. It is fast by any biological standard at these depths, and it leaves behind something that the next stage will find transformed almost beyond recognition. When the last of the large scavengers withdraws, what remains on the seafloor is a skeleton. It is not a bare skeleton, not yet. There are patches of remaining soft tissue in recesses the large scavengers could not easily reach. Connective tissue, cartilage, remnants of organ packed into the body cavities of the spine. The sediment immediately around the bones is rich with organic material that leached from the body during and after the scavenger stage, saturated with lipids that the whale stored in its tissues during its years of life at the surface. The bones themselves hold more. The cancellous matrix of a baleen whale's vertebrae and ribs is riddled with microscopic channels, and those channels are packed with pale, wax-like lipids, reserves of chemical energy so dense that they persisted in the bone for the entire life of the whale, held against need, never depleted, and still present now. The sediment darkens around the skeleton in the weeks following the withdrawal of the mobile scavengers. It is a visible change, detectable to cameras and to the instruments of the researchers who have returned to monitor these sites over years. The darkening comes from the organic loading, lipids, and smaller organic compounds seeping from the bones and mixing with the gray seafloor sediment, changing its texture and chemistry in a spreading halo around the whale's remains. Within this halo, the second stage of succession begins. The enrichment opportunist stage takes its name from what drives it, an extraordinary and temporary richness in the substrate, where the background deep seafloor receives only the slow, diffuse fall of small organic particles drifting down from the sunlit surface, a sparse input, thin and dilute, spread across the entire ocean floor. The sediment immediately surrounding the whale skeleton has become saturated with concentrated organic material. The difference in energy density is not a small one. The sediment within a meter of the bones may hold hundreds of times the organic carbon present in the surrounding seafloor. For organisms capable of colonizing this enriched substrate, it represents a temporary but extraordinary concentration of resources. What colonizes it first is small and dense. Polychaete worms, bristleworms, the ecologists' familiar deep-sea generalists, arrive in numbers that are difficult to picture. Mat-forming species capable of tolerating high organic concentrations move into the sediment around the bones. They are followed by small crustaceans, by mollusks, by the whole opportunist fauna of the deep-sea enrichment community, organisms that have evolved to exploit exactly this kind of temporary organic bonanza wherever it occurs on the seafloor. An oil spill, a sunken log, a clump of kelp that drifted down from the surface, a whale. Each of these is a different scale of the same kind of event. Concentrated carbon arriving at the seafloor and being consumed by whatever is adapted to consume it quickly. At a whale, fall the concentration is simply larger, and the opportunist community it supports is proportionally denser. Population densities in the enrichment community at documented whale fall sites have reached hundreds of thousands of individuals per square meter of sediment, numbers that belong to the richest shallow water environments, brought into being in the deep abyss by the chemistry of a single death. The bone surfaces are not empty either. While the sediment community proliferates in the halo around the skeleton, the surfaces of the bones themselves are being colonized. The organic remnants still adhering to the bone, connective tissue, lipid-saturated periosteum, the residue of soft tissue that the large scavengers left behind, are broken down by smaller organisms that the earlier mobile scavengers had no use for. Small crustaceans pick at the bone surfaces. Polychaetes thread into recesses between the ribs. The bone begins to be furred with microscopic life. Early colonizers, whose feeding activity is physically cleaning the outer surface of the skeleton, while the bone interior remains sealed and saturated with stored lipid. There is a patience to this stage that becomes apparent only over time. Nothing here is hurrying. The organisms of the enrichment community do not deplete the organic reserves around them in days or weeks. The sediment halo is thick with material, and the bone surfaces offer months of feeding for the organisms that have settled there. Surveys of documented whale fall sites in the years following carcass arrival show the enrichment community persisting for months to several years, gradually shifting in composition as the most accessible organic fractions are consumed first, and the slower releasing ones remain. The transition from the enrichment stage to the third stage is not a sharp line. It is a gradual deepening, a shift in the chemistry of the site that happens from the inside out, beginning within the bone matrix, where the saturation of lipids and the exclusion of oxygen from the most protected internal channels creates conditions for a different kind of biology entirely. The third stage is the long one. It is called the sulfophilic stage, because what drives it is the production of hydrogen sulfide by anaerobic bacteria inside the whale bones. Within the lipid-saturated cancellous matrix, where oxygen cannot penetrate and the organic concentration is immense, sulfate-reducing bacteria conduct a form of respiration that uses dissolved sulfate from the surrounding seawater as an electron acceptor. The product of this respiration is hydrogen sulfide, a gas in higher concentrations, but here in low continuous concentrations, a chemical that diffuses outward through the bone and into the surrounding water, creating the foundation for an entirely different kind of food web. The shift is chemical, but its biological consequences are vast. Hydrogen sulfide diffusing outward from the whale bones reaches the bone surface and the immediately surrounding sediment and water, and there it encounters a group of organisms that evolve to treat it not as a waste product or a poison, but as food. Sulfur oxidizing bacteria, including the filamentous forms of the genus Bigiato, colonize the bone surfaces and adjacent sediment and build mats that in favorable conditions grow centimeters thick. These bacteria conduct a form of primary production analogous to photosynthesis. They fix carbon from the dissolved inorganic carbon in the seawater using the chemical energy of sulfide oxidation, producing organic matter from inorganic materials without any need for sunlight. At 2 kilometers below the surface, in absolute darkness, in water where no photon from the sun has ever arrived, there is primary production. There is new organic matter being manufactured. There is, in the fullest biological sense of the word, a food web built on chemistry rather than light. This is the longest of the 4 stages. The lipid reserves inside a large baleen whale's skeleton are vast. The cancellous matrix of the vertebrae, the ribs, the skull bones, the limb bones reduced and fused in the whale's evolutionary history from the walking legs of its terrestrial ancestors, all of these hold oil, and all of it must be consumed before the sulfide system begins to fail. Estimates of the time required span decades. A large blue whale skeleton may sustain the sulfophilic community for 50 years or more. There is, within the bones of a single animal, enough chemical energy to maintain an entire ecosystem for half a century. The organisms that colonize the sulfophilic site are not the same as those of the enrichment stage. They are specialist fauna, organisms that have evolved specifically for this kind of chemosynthetic habitat, the same kinds of organisms, and sometimes the same genera and families found at hydrothermal vents and cold seeps in the deep ocean. Chemosynthetic mussels settle at the base of the whale vertebrae, their gills packed with sulfur oxidizing bacterial symbionts that manufacture the mussel's nutrition from the same sulfide chemistry powering the bacterial mats. Snails graze the mats, worms thread the sediment, and among the most striking colonists of all, anchoring themselves directly to the bone surfaces and boring into the bone matrix with root-like structures, are the bone-eating worms of the genus Osedax. But these are the details of a single stage. The details belong to the stage itself, and there will be time to settle into them. What matters here, in this overview of the succession's full arc, is the scale of what the sulfophilic stage represents in the economy of the deep sea floor. The surrounding abyssal sediment, in the absence of the whale fall, receives perhaps a few milligrams of organic carbon per square centimeter per year, arriving as marine snow, the slow, diffuse rain of small particles drifting down from the productive surface waters above. The whale fall site, during the sulfophilic stage, is receiving no new organic carbon from the surface. What it is doing is recycling the carbon stored in the whale's bones over the whale's entire life, releasing it slowly through microbial processing, and using it to support a community of hundreds of species at densities the surrounding seafloor will not see again for a very long time. The whale fall is not receiving, it is giving, and it will keep giving for decades, measured not in the slow years of a human career, but in the generational time of the organisms living at the site. Species whose entire life cycles, from larval settlement to reproductive maturity to death, play out against the backdrop of slowly diminishing sulfide concentrations and the gradual exhaustion of the bone lipids. When the lipids are gone, the sulfide production slows, the bacterial mats thin. The specialist chemosynthetic fauna that depended on the sulfide gradient begin to lose the chemical foundation of their food web. This is a slow process too, not a sudden collapse but a gradual withdrawal, the community contracting over years as the energy source diminishes. The organisms that cannot persist without the high sulfide concentrations begin to disappear from the site. The organisms that were less dependent on the sulfide chemistry, the more generalist species that had been present at the edges of the whale fall community during the richer stages, are proportionally more represented in what remains. What remains, finally, is a structure, a skeleton. The fourth stage is built on what the first three stages have left behind. The whale bones, by the time the sulfophilic community has exhausted the lipid reserves, have been transformed. The outer surfaces have been scoured and etched by decades of biological activity, by the boring structures of Osedax worms, by the grazing of snails and amphipods, by the slow acid dissolution of the bacterial communities. The cancellous interior, once packed with pale lipid-soaked tissue, is now largely empty, a honeycomb of channels that no longer hold reserves, open to the seawater that percolates through them from outside. The bones are lighter than they were, they are more porous, but they remain standing or lying on the seafloor, as they have for 50 years or more. And in their transformed state, they offer something, hard substrate. On the deep abyssal plain, hard substrate is rare. The seafloor for hundreds and thousands of miles in every direction is soft sediment, gray mud, The accumulated fall of fine particles from the surface over millions of years, deep and yielding, and offering no purchase for organisms that need a firm surface to attach to. Corals cannot grow in sediment. Sponges cannot anchor in mud. The suspension feeders that build their bodies on hard substrate, and there are many of them, ecologically important, architecturally elaborate, have nowhere to settle. A whale skeleton, even one stripped of its organic content, is hard. It is calcium phosphate and protein, far denser and more resistant than any particle of marine snow, and it will persist on the seafloor for years after the last of the sulfite chemistry has faded. Corals settle on the bone surfaces, encrusting invertebrates spread across the ribs and vertebrae. Feather stars anchor their flexible arms to the curved arch of what was once a whale's jaw. Sponges grow from the rounded surfaces of the skull, the skeleton that was once an ecosystem of scavengers, an ecosystem of opportunist enrichment, an ecosystem of chemosynthetic production, becomes in its last ecological phase something more familiar, a rocky outcrop, a hard reef, a permanent piece of structural complexity on the otherwise featureless floor. This is the reef stage. It is not dramatic. It does not support the extraordinary species density of the sulfophilic stage or the massive biomass of the mobile scavenger stage. It is quieter, slower, a gradual integration of the whale bones into the surrounding benthic community, the skeleton becoming less and less distinguishable over the years from any other hard substrate on the seafloor. The animals that settle here do not know they are on whale bones. They know only that the surface is hard and stable, and that the currents carry food particles past this place, and that they can attach and filter and grow. In time, the whale fall becomes part of the landscape. The sediment creeps back. Fine particles settle on the horizontal surfaces of the bones, the upward-facing ribs, the flat plates of the skull. The encrusting organisms grow over the bone surfaces and trap sediment in their structures, gradually burying the outermost edges. The whale skeleton is not gone. It will persist for a very long time, but it is becoming less a distinct sight and more a feature of the benthic community that surrounds it. A hard structure among other structures, colonized and occupied and continuously inhabited, no longer as the temporary and extraordinary resource it was for the first decades, but as a permanent and quieter part of the deep sea floor. And still, if the bones could be excavated from their encrusting communities and examined, they would show the marks of everything that came before, the boreholes of Ozadax worms, threading down into the cancellous matrix from dozens of points on each bone surface, the scour marks of early scavengers, the pitting and etching of bacterial activity across decades. The whale fall's entire history is written in the bones. If the reader has the patience to look, the complete arc spans time that is hard to feel all at once. From the moment the carcass settles to the moment the skeleton is fully integrated into the reef stage, a large baleen whale fall may encompass 70, 80, perhaps 100 years. The mobile scavenger stage ends in months. The enrichment stage lasts months to years. The sulfophilic stage alone may run for 50 years or more in the most lipid-rich skeletons, and the reef stage, which has no clear ending, continues for as long as the bones persist, a century, at a single site on the seafloor, from a single death. To put that in a different frame, a whale that died in the deep ocean at the beginning of the 20th century, if its carcass came to rest at the right depth and water, cold enough and still enough, might still be supporting a chemosynthetic community today. The hydrogen sulfide still diffusing outward from bones 100 years old. The bacterial mats still growing on bone surfaces exposed to the water. the whole apparatus of a chemosynthetic food web still operating in complete darkness from a death that occurred before almost every living human was born. There is no urgency to any of this. That is perhaps what is most striking about the whale fall succession when the full arc is laid out at once, the almost geological patience of it, the way each stage operates at its own temporal scale, the way time at the seafloor is simply different from time at the surface. A community that would take a century to fully cycle through its stages is not exceptional in the context of deep-sea biology. The deep ocean is old in ways that the productive, turbulent surface is not. The organisms that live here have evolved for conditions that change slowly, and the whale fall, arriving on the seafloor as something extraordinary and temporary, is processed slowly, fully, with none of the urgency that scarcity might impose closer to the surface. The pace of the succession slows as the stages deepen. The mobile scavenger stage is fast, almost frantic by deep-sea standards. The large scavengers moving in and withdrawing, the tissue disappearing month by month. The enrichment stage is slower, colonizers settling into a substrate that will sustain them for a year or more. The sulfophilic stage is the slowest of all the active biological phases, its 50-year span making it longer than most human careers in science, longer than the entire span of deep-sea research using remotely operated vehicles. And the reef stage has no end that can be predicted. The succession is also not entirely sequential. The stages overlap. The sulfophilic community does not wait for the enrichment community to completely clear before it establishes itself. The first sulfide begins to diffuse from the most protected bone surfaces, while the enrichment community is still processing the outer layers of the skeleton. The reef stage community begins to establish itself on the parts of the skeleton where the lipid reserves were shallowest and the sulfide production ended earliest, while other parts of the skeleton are still supporting chemosynthetic biology. The 4 stages are a framework, a map, not a rigid sequence of clean transitions. Real whale falls are messier, richer, more layered than the framework suggests. What the framework gives is orientation, a way of understanding how a single death can support 4 ecologically distinct communities across a span of time that extends beyond any individual human observation. The researchers who first documented whale falls in the late 20th century did not live to observe the full succession of the sites they discovered. The scientists monitoring those sites today are the professional descendants of the original researchers, and even they may not observe the complete exhaustion of the sulfophilic stage they are currently studying. The whale fall outlasts the scientist's career. It may outlast several. This is one of the reasons the deep ocean keeps its secrets so well. A surface ecosystem, a meadow, a forest, a reef, can be observed across its full successional arc within a human lifespan, sometimes within a decade. The deep-sea whale fall cannot. It requires a commitment to monitoring that outlasts the individual researcher, a passing of knowledge from generation to generation of deep-sea scientists, a continuity of attention that mirrors in some ways the continuity of the succession itself. And even with that continuity, the observations are sparse. There are perhaps a handful of documented whale fall sites that have been repeatedly visited by remotely operated vehicles over years or decades. The deep ocean is vast. The whale fall sites within it are rare and scattered. The chance of a research vessel encountering a natural whale fall is small, and most of what is known about the later stages of succession comes from a small number of monitored sites and from experimentally implanted carcasses, whale bodies that died in circumstances where their carcasses could be retrieved and brought to known locations on the sea floor, then revisited. The science of whale falls is young in the way that all deep sea science is young. The technology to visit these places reliably has existed for only a few decades. The framework of 4 successional stages was assembled from observations made across those decades, and there are still questions within each stage about the timing of transitions, about the role of whale species identity and body size in shaping the succession, about the geographic variation in whale fall communities across different ocean basins that remain open. The framework is solid, the details within it are still being filled, and the succession itself continues, indifferent to the pace of human understanding, playing out at sites distributed across the deep sea floor in every ocean basin at depths ranging from a few hundred meters to several thousand, wherever a whale has died and its body has reached the seafloor intact. In the North Pacific, in the South Atlantic, in the Southern Ocean around Antarctica, where whale populations were once among the densest on Earth, and commercial hunting reduced them to fractions of their former numbers. In every basin where the deep sea floor receives the weight of what was once the largest animal alive. The plume of dissolved molecules that began the succession has long since dissipated into the background chemistry of the surrounding water. The skeleton that generated it is still there, still giving. The bacterial mats may still be growing on bone surfaces that have not seen light since the whale was carried to this depth. The slow current that passes over the site carries hydrogen sulfide outward at concentrations too low to detect without instruments. And at those concentrations, it feeds bacteria that feed worms, that feed snails, in a food web that needs no sunlight, no surface, no connection to the productive layer of the ocean above. The 4 stages arrive and recede like seasons measured in decades. The first one comes on fast and warm, the large organisms moving in and filling the site with the most biological activity it will ever see. The second one settles in more quietly, the sediment darkening, the small organisms packing the substrate around the bones in dense and patient populations. The third one unfolds so slowly that a researcher returning to the site 5 years after their first visit might find it apparently unchanged. The same bacterial mats, the same community of specialist organisms, the same hydrogen sulfide diffusing from the same bones, and yet from within the bone matrix, the lipid reserves are steadily diminishing, almost imperceptibly, the way a very thick candle burns all night, with no apparent change, but is shorter in the morning. The fourth one is the quietest, the most ordinary from the outside, the encrusting organisms growing over the bones in the slow way of all sessile deep-sea life, turning the site from a temporary anomaly into a permanent fixture of the benthic landscape. Through all 4 stages, the bones endure. They are the constant, the element of continuity across 100 years of change. Everything else comes and goes, the sharks and hagfish of the first months, the dense polychaete mats of the enrichment community, the bacterial mats and chemosynthetic mussels of the sulfophilic stage, the corals and sponges of the reef phase. They arrive, they process what the site offers, they disappear when the offering is exhausted or superseded. The bones remain, and within the bones, diminishing year by year, decade by decade, the last of the lipids, the stored oil of a whale that fed and swam and breathed and dove at the surface, that accumulated this chemical wealth through years of feeding on krill and small fish in the productive surface waters, that carried it downward in death and in bone all the way to this place, this cold, dark, still, pressurized place where the seafloor has been receiving the slow fall of marine snow for longer than any whale has existed, is being consumed, gram by gram, by organisms too small to see, in the service of a food web too deep to visit easily, for a period of time too long to watch in a single career. The succession does not announce its end. There is no threshold crossed, no signal sent. The lipids simply diminish until the concentration within the bone matrix can no longer support the sulfate-reducing bacteria at rates sufficient to maintain the sulfide gradient, and the bacterial mats thin, and the chemosynthetic community contracts, and what remains is cleaner and quieter than what was there before. And then the reef community persists, and the bones endure beneath their encrusting organisms, and the site continues. It continues for a very long time. In the deep ocean, a site that has been an active whale fall for a century becomes part of the seafloor's biography, a record of what happened here, encoded in the pitted and bored and encrusted bones, available to anyone who visits with the right instruments and the patience to read what the bone surface shows. The deep sea is a slow archive. Nothing moves quickly enough to erase the record before it is set. The whale fall succession is, from one angle, a decomposition, the processing of a body, the return of its stored carbon and nutrients to the water column and the sediment. From another angle, it is something closer to a generation, a long, patient creation of habitat and community and biological diversity from a single event, a single death, on a seafloor that would otherwise be nearly empty. Both of these things are true, and neither cancels the other. The cold water moves across the site in the same slow current it has always moved. The bones rest in the sediment as they have rested for decades. In the darkness above the seafloor, 2 kilometers of cold water separates this place from the surface and the sun. And at the site itself, in the slow and patient way of the deep, something is still living. Something is still living from the whale. It will be living for a long time yet. The mats still growing, the worms still boring, the encrusting organisms still spreading across the last surfaces of the old skeleton. The whole slow transformation still unfolding through stages that began decades ago and have not yet reached their quiet end. The succession continues through its phases with the same unhurried inevitability as the marine snow that falls above it, particle by particle, season after season, in an ocean that has been receiving the dead since long before there were whales to fall into it. The skeleton rests on the sea floor in its long dark stillness, and the century of life it carries is patient and complete and ongoing. In the deep water, far from any shore, a scent moves through the dark. It has no color and no form. It drifts outward from a single point on the sea floor in a slow expanding thread, a trail of dissolved amino acids and lipid compounds carried by the faint bottom currents, thinning as it travels, growing fainter with each kilometer it covers, but never entirely disappearing. Cold and dark, and carrying the precise molecular signature of something very large that has recently come to rest below. The water around it is still. The pressure at this depth would collapse a human lung to the size of a fist. Nothing down here burns a light. A kilometer away, a pale shape moves through the darkness. It moves slowly. Nearly everything in this water moves slowly, as if the cold and the pressure have set a speed limit that living things long ago learned to obey. The shape is large, wider at the front and tapering toward the rear, and it angles gently downward as it travels, following the gradient of the scent, the way a hand trails along a wall in a dark room. It does not hurry. It does not need to. What it is following has not moved and will not move, and the distance between them is closing, one slow body length at a time. It is a Pacific sleeper shark, and it has turned toward the source of the scent in the way that its kind have been turning towards scents like this one for longer than our species has existed. The Pacific sleeper shark is not built for speed. It belongs to the same family as the Greenland shark, and like that animal, it moves with a patience that seems almost geological, a slow, continuous glide through cold water, barely visible to the lights of any camera that might be trained on it, its pale belly faintly luminous in the absolute dark. It can reach lengths of over 14 feet. It has been found at depths beyond 6,000 feet. It carries no sense of urgency in the way it moves, because in the world it inhabits, urgency is rarely necessary. Food in the deep sea is scarce and often comes in small increments, a fragment of organic material drifting down from above. A small fish, a piece of something that was once alive but is now only chemistry drifting in the current. A whale is different. A whale is something else entirely. The scent plume is thicker now, denser, and the shark follows it without deviation. Below, the carcass rests on the sea floor in a stillness that has not yet been broken. The soft tissue is intact. The blubber layer along the flanks, pale and dense, carries enough energy to sustain a large predator for months. The muscle beneath it is cold and dark red and holds within its fibers the stored chemistry of years of feeding in sunlit surface waters. The bones below the muscle are saturated with lipid, a waxy, oil-rich material packed into the spongy interior channels of every vertebra, every rib, filling the skeleton the way water fills a sponge. The total weight of the animal, even stripped of seawater, is tens of thousands of pounds. It has come to rest on the fine gray sediment of the bathyal floor, and the sediment has barely yielded beneath it. The carcass is enormous and entirely still, and the water around it has already begun to carry its signal outward in every direction, broadcasting through the cold dark to any organism with the chemical sense to read it. The shark arrives and does not pause. It moves along the carcass's flank, close enough that its skin brushes the blubber, and then it does something particular and practiced. It turns its body broadside to the whale's side, opens its jaws against the blubber layer, bites down, and rotates a slow, torquing roll of its entire body that uses its own weight and the resistance of the carcass as opposing forces, and with that leverage tears free a section of blubber several pounds in weight. The motion is unhurried but efficient, the result of a body plan refined over hundreds of millions of years for exactly this kind of dense, resistant tissue. The shark withdraws, swallows, and returns. There is no competition yet. The scent plume has been traveling for hours, and the deep sea is wide, and what arrives first must come from nearest. Other shapes are coming. In the water column above the sea floor, rising and falling in slow arcs, a group of rat-tail fish catches the scent below. These are long, tapered animals, broader at the head, narrowing to a thread-thin tail that gives them their name, and they are among the most abundant deep-sea fish in the ocean. Any square mile of abyssal seafloor in the world's major ocean basins contains rat-tail fish, hovering in the mid-water or cruising close to the bottom, feeding on whatever organic matter the slow drift from above occasionally delivers. They are opportunists, but they are persistent ones. They angle downward now, their blunt heads leading, their long tails streaming behind them in the still water, descending toward the floor and the whale lying on it. At the bone surface, smaller things are already working. The hagfish arrived first, or nearly first, cylindrical, nearly featureless in the dark, lacking the jaws of the shark or the teeth of the rat-tail fish, but lacking almost nothing else of consequence. They have no bones and no scales and secrete a thick gel from their skin when disturbed, a substance so dense and slippery that it can clog the gills of any predator that tries to take them. They are not fast. They do not need to be fast. What they have instead is a set of rasping teeth on a flat protrusible tongue, and the patience to use them indefinitely. A hagfish pushes itself directly into the soft tissue of the carcass and begins to feed from the inside, boring into the muscle and working its way deeper by a slow, writhing movement of its entire body. It can absorb dissolved organic molecules directly through its skin while it burrows, feeding at the cellular level even before its rasping teeth have reached anything solid. The carcass, viewed from outside, still looks largely intact. Inside, the hagfish are taking it apart, one tunnel at a time. There are dozens of them. More arrive. Hagfish navigate by chemical sense in water, where light has never penetrated, and the plume of dissolved compounds emanating from the whale is a signal they can follow with the same precision as the shark that preceded them. They arrive from multiple directions, converging on the carcass from the surrounding seafloor like slow threads drawn toward a center. Each one finds an entry point, a natural opening, a wound opened by the shark, a soft region where the skin has loosened from the underlying tissue and pushes inside. The water around the carcass begins to take on the faint shimmer that comes from concentrated dissolved organics, a barely visible distortion of the ROV lights, as if the water itself is becoming richer and more complex, absorbing the slow chemical exhalation of the whale. The shark rolls again, removing another section of blubber from the flank. By now, a second and third shark have arrived. They move in loose arcs around the carcass, not coordinating with each other, but not interfering either, each one oriented toward the whale, each one taking its approach when the others have withdrawn, the rhythm of feeding establishing itself, with the informal spacing of animals that have learned, through long experience in a world where food is rare, that patience is its own strategy. Two sharks feed at the same site, and then one withdraws, and the other continues, and a third drifts in from the far side, where the blubber on the dorsal surface has not yet been touched. A column of amphipods descends from the water above. They are small, each one no longer than a thumb. They belong to a group sometimes called Lycian acciped amphipods, scavenging crustaceans that spend much of their time drifting in the mid-water column, rising and descending with the slow vertical currents, waiting for exactly this kind of signal. When it comes, they orient toward it and swim, and because there are hundreds of thousands of them in the water column above any given stretch of deep sea floor, the signal from a whale carcass does not have far to travel before it finds them. They arrive as a soft, pale cloud, settling over the exposed muscle where the sharks have torn away the blubber, and they begin to feed. Each individual removes a tiny fraction of tissue per minute. Together, moving in a dense wave across the exposed surface, they process the whale at a rate that rivals the output of animals 1,000 times their individual size. The rat-tail fish arrive and hover. They descend to within a body length of the seafloor, orienting to the carcass, and then they approach. They feed at the torn edges of the blubber, at the exposed muscle surfaces, pulling tissue away with their mouths, and then withdrawing, drifting up a few feet, and then descending again. They feed in bursts, and between bursts they hover with their long tails hanging below them, waiting as if conserving energy for the next approach. In the ROV footage that researchers have gathered at sites like this, footage compressed into time-lapse sequences, hours becoming minutes, the rat tails look like attendants at something ceremonial, their slow up and down movement giving the scene a rhythm that feels almost deliberate. Though nothing here is deliberate, only efficient. The night does not change because at this depth there is no night. There is only the dark which is constant, and the cold, which is constant, and the slow removal of tissue from the whale, which continues without pause. By the second day, the visible injuries have accumulated. Large sections of blubber have been removed from the flanks and the dorsal surface. The exposed muscle beneath is pale, where the cold has worked on it, and darker where feeding has been most concentrated. The sharks continue to visit, arriving and departing in no fixed pattern, following the plume inward, and then withdrawing to process what they have taken. The hagfish are still boring deeper into the carcass, their tunnels now opening the interior to seawater, accelerating the chemical changes inside the body in ways that will matter for what comes much later. The amphipods work continuously, their feeding never stopping, their collective removal of tissue measured in pounds per hour when their density is high enough. The scent plume extends further now. As tissue is removed and fluids are released, the chemical signal grows stronger rather than weaker, and it spreads wider across the seafloor and into the water column above. Organisms that were beyond sensing range 2 days ago can detect it now. New arrivals appear, more rat-tail fish, more amphipods, a second species of hagfish, various small invertebrates drawn by the disturbed sediment, and the organic particles settling from the edges of the feeding site. The whale that was invisible on the seafloor, an isolated object in an otherwise empty landscape, has become a center. Around it, the normally vacant sediment is becoming a different kind of place. A sleeper shark feeds at the base of the tail. It has found a region where the tissue is dense and the blubber layer thick, and it works methodically, rolling and tearing, rolling and tearing. The motion is slow enough to watch without strain. There is no violence in it, nothing that reads as aggression, only the precise physical operation of a jaw and a body against resistant material. Blubber is composed largely of lipids, the same class of compounds that will eventually power the ecosystem for decades from within the bones. And right now, the sharks are removing the most accessible fraction of that energy store, the outer layer, the part most exposed and most easily processed. What remains, the deep bone lipid, the saturated fat held inside the porous skeleton, is beyond their reach and beyond the reach of every large scavenger gathered here. The bones will be reached by other means, in other time. The sediment around the carcass begins to change color. Lipids and organic fluids leaking from the torn tissue have begun to saturate the fine gray sediment immediately surrounding the whale, darkening it slightly, altering its chemistry in a way that will persist long after the carcass itself has been transformed. The halo of affected sediment is perhaps 3 feet wide in the earliest stages, a thin ring of enrichment around the bare bones of the whale, barely visible but chemically significant, the first evidence that the impact of this whale on this patch of deep-sea floor extends beyond the footprint of the body itself. The weeks pass, nothing here counts them. Time at this depth is not marked by light or temperature or the seasonal shifts that organize life in the shallower ocean and on land. It is marked only by the change in the carcass, by what has been removed and what remains. By the end of the 3rd week, the bulk of the blubber on the upper surface and the flanks has been stripped away. The exposed muscle is cooling further as the insulating layer is gone, and the cold seawater now contacts the tissue directly, slowing the microbial decomposition within, but also altering the texture of the exposed surfaces in ways that make them more accessible to the smaller organisms. The shark visits are still regular, but the largest and most accessible portions of tissue are now gone, and the sharks spend more time circling before finding a productive feeding site. The amphipods are everywhere. They coat the exposed muscle in a moving carpet, each individual working independently. the collective movement giving the surface a texture like something breathing, where the tissue has been exposed longest and cooled most completely. The amphipods feed most densely, their small bodies packed together at a concentration that would be difficult to believe if it had not been documented on footage gathered by the very researchers who first described what a deep-sea whale fall becomes. Hundreds of thousands of individuals per square foot are not an exaggeration. They are a fact recorded in field observations, a density of life that concentrates here, because here, in the deep ocean, is where the food is. Small crabs have found the site. They move across the sediment with the purposeful horizontal motion of crustaceans, their legs picking carefully across the disturbed substrate, their claws working at the smaller tissue fragments that have come free and settled around the base of the carcass. They are thorough. Nothing in the deep ocean survives by being wasteful of energy, and any organism that makes the journey to a whale carcass has been drawn by a signal strong enough to justify the cost of travel and will not leave until the resource is exhausted or the competition for it becomes prohibitive. The crabs work the margins, while the sharks work the flanks, and the amphipods work the surface, and the hagfish work the interior, and between them they occupy nearly every available feeding niche simultaneously, a division that no organism planned and none is aware of. But that emerges from the simple fact that each organism follows its own best strategy in a place where food is suddenly, temporarily, abundant. 2 months have passed, perhaps a little more. The whale is not gone. Not yet. The soft tissue is largely removed from the upper surfaces and the flanks, but the lower side, the surface resting against the sediment, has been slower to expose. Some tissue remains in the deeper recesses of the body cavity, and the hagfish have carried tunnels through much of what was the abdominal region, leaving a landscape of hollow spaces within the carcass that the current and the smaller organisms are still working through. The skeleton is becoming visible in places where it was not visible before. The long curved lines of the ribs emerge from the remaining tissue, like the hull of a ship being exposed by the slow tide of the feeding. They are pale and clean where they have been exposed and still dark with tissue where they have not. They curve upward from the seafloor and then arch back down, and the whole structure is still recognizably a whale, still carrying the shape of the animal that was here, even as that shape is being steadily undone. A large rat-tail fish descends to the exposed rib surface. It noses along the bone, finding a fold of remaining tissue caught in the angle where the rib meets the vertebra, and feeds there for several minutes before withdrawing. Around it, the amphipods continue their steady work on the exposed muscle. The shark that has been circling the carcass for 2 days descends again to the lower flank, where a seam of blubber has been exposed by the slow deflation and settling of the body as the tissue above it was removed. It rolls, it tears, it withdraws. The seafloor has been transformed within a radius of perhaps 20 feet in every direction. The sediment is darker here than it is beyond the halo. Organic particles have settled from the feeding activity, from the fragments of tissue and the dissolved compounds that have diffused outward from the carcass, and they have altered the microbiology of the sediment itself, enriching it with carbon, changing the redox chemistry, beginning a process of stratification between the surface sediment that remains in contact with oxygenated seawater and the deeper sediment below, where oxygen is being depleted by the sudden abundance of organic material. The seafloor around the whale is no longer what it was before the whale arrived. It will not return to what it was before for a very long time. By the 3rd month, the character of the scavenger community is changing. The largest animals are visiting less frequently. The sharks, which were the dominant presence in the earliest weeks, appear in the footage with less regularity. The resource they were best equipped to exploit, the thick blubber, the dense outer muscle, has been largely consumed, and what remains is less accessible to them. They are moving on, following other scent plumes across the deep sea floor to other opportunities, carrying within their bodies the chemical energy that was stored in the whale's tissue and will now be metabolized, spent, distributed across a wide area of the ocean floor as the sharks resume their slow patrol of the dark. What they carried away from this place is gone from here, but not lost. It continues, dispersed, in the bodies of animals that will themselves eventually die and feed other things. The amphipods are thinning. Where they were dense enough to coat every exposed surface 2 months ago, they now concentrate at the sites where tissue remains. Their numbers are still high. There is still tissue here, still chemistry worth consuming, but the peak is past, and the feeding frenzy that the early weeks represented is settling into something slower, more deliberate. Smaller organisms are becoming more prominent. Polychaete worms have appeared at the sediment surface around the base of the carcass, threading through the organic-rich substrate, consuming the particles and the dissolved compounds that have settled there. Tiny snails move slowly across the exposed bone surfaces. The community is shifting in its composition. The species best suited to consuming large quantities of accessible tissue giving way to the species best suited to processing what remains. The bones are almost fully exposed on the upper surface now. Months of feeding have cleared the soft tissue from the dorsal and lateral surfaces of the skeleton, and the full architecture of the whale is visible. The long series of vertebrae running from the skull to the tail, the ribs arching outward from the spine and curving back down to their ventral endpoints, the massive bones of the skull lying separate from the body and embedded partly in the sediment where the head came to rest. The bones are clean, where the scavengers have worked most thoroughly, and still carrying a thin, adherent layer of tissue where they have not. They are pale, almost white in the ROV light, the waxy pallor of bone from which the overlying tissue has been stripped, but which still retains its lipid-saturated interior, hidden inside its own matrix, not yet released, not yet available to the organisms that will come next. The skeleton has the quality of something revealed, not created. It was always there, inside the whale, and now it is simply bare. The form of the animal persists in the arrangement of the bones, even as everything that made it move and breathe and swim is gone. The curve of the rib cage still holds the approximate volume of the body cavity that once contained the heart and the lungs. The individual vertebrae, each one wider than a human torso in a large whale, still carry the articulating surfaces where they met each other in life, the smooth concave faces that allowed the whole long spine to flex and drive the animal through open water. The bones are, in a real and physical sense, what remains of the whale, and they contain more of the whale's stored energy than all the tissue that has already been consumed. The scent plume is different now. In the earliest weeks, it was rich with the compounds of fresh, soft tissue, amino acids, proteins, lipids from the blubber. Now the profile of the plume is changing as the source material changes. The soft tissue is largely gone, and what the water carries from this site is shifting toward the compounds that emanate from the exposed bone surface, the lipids seeping slowly outward from the cancellous matrix, the dissolved products of microbial activity beginning within the bone itself. It is a quieter signal, thinner, more diffuse, less urgent, and the organisms it attracts in the months to come will be different in kind from the scavengers that answered the first, louder call. For now, though, the scavengers are still here. A small group of amphipods moves across the ventral surface, where the carcass has pressed against the sediment for 3 months, and the tissue on that side has been slower to clear. They work with the same steady collective patience they have maintained since the earliest days of the feeding. No urgency, no hurry, only the continuous incremental removal of material from a surface that will eventually be clean. A single rat-tail fish descends, noses at the bone surface, finds nothing accessible, and rises again. The water directly above the skeleton is slightly warmer than the surrounding seafloor water by a fraction of a degree, the heat of microbial metabolism within the bones already beginning to alter the local chemistry in ways too subtle to see, but not too subtle to measure. 4 months have passed since the whale came to rest. The estimate made by the researchers who first carefully studied sites like this one, that a well-attended deep-sea whale carcass might lose 90% of its soft tissue within 4 months, is borne out here in what is visible and what is not. The whale is a skeleton, not a bare one, not yet entirely clean, but a skeleton nonetheless, the form of the animal rendered in pale bone on the flat gray sediment, the ribs arching and the vertebrae running and the skull enormous and settled and still. The sediment around it is dark with organic enrichment. The microbial populations within the bone are already beginning the slow work that will sustain this place for decades more. The shark that fed here on the first night is long gone. It took what it needed and moved on, carrying within its body the chemical energy of the whale's outer layers, dispersed now across miles of deep-sea floor. The hagfish have retreated from the hollowed interior of the carcass and moved away to other opportunities, or settled into the sediment nearby, where the organic enrichment sustains them in the interim. The rat-tail fish are present only occasionally now, visiting less from appetite than from the persistent trace of scent that the bones continue to release. The amphipods are reduced to a small, steady presence along the bone margins and the enriched sediment immediately surrounding the skeleton. The carcass rests in a stillness it has not experienced since it first came to rest. Not the stillness of absence. The bones are not empty. The sediment is not bare. The chemistry within the skeleton is active and continuing and will be active and continuing for a very long time. But the drama of the early months, the sharks rolling against the blubber, the hagfish boring into the soft tissue, the dense waves of amphipods moving across every exposed surface, that phase has closed. What the scavengers did here was absolute and efficient and swift, measured against the time scale of what comes next, and now the skeleton lies pale and quiet on the seafloor in the continuous dark, the curved forms of the ribs still holding the shape of something that once breathed. The water around the bones is still not silent. There is no real silence in the ocean, which carries sound across vast distances through its pressure waves and its chemistry, but still in the way the deep sea is always still, the currents barely perceptible, the temperature nearly constant, the light entirely absent. The skeleton does not move. The sediment that was disturbed by months of feeding activity has begun to settle again, the fine particles drifting back down to coat the disturbed areas in the thin, even layer that the deep ocean floor is always, slowly receiving from above. What the scavengers consumed is gone, distributed through the bodies of hundreds of species across hundreds of miles of the deep ocean floor. What they left behind is everything. The skeleton holds, in the saturated channels of its bone matrix, more stored chemical energy than the entire mass of soft tissue that has just been consumed. It is packed into the lipid-rich, spongy interior of every bone, The vertebrae, the ribs, the skull, the long flat scapulae, a reserve of stored oil that will take not months but decades to exhaust. The organisms that will access it will not be sharks or hagfish or amphipods. They will be smaller and stranger and built for a longer, slower chemistry. They are not here yet. The bones are not ready for them yet, in quite the right way, for now the skeleton rests clean and quiet and pale in the dark grove. Having fed an entire guild of scavengers through 4 months of continuous activity, and it holds within its structure more chemistry than it has yet released, more life than it has yet sustained, more time than the animals that consumed the soft tissue will live to see. The deep sea floor stretches away from it in every direction, flat and featureless and gray, and the skeleton lies at the center of a small halo of darkened, enriched sediment that marks the radius of its influence, and the water above it carries the faint, slow signal of the lipids beginning their long journey outward from the bone. The ribs curve upward in the dark. They are still and pale and full. From the surface of a whalebone resting on the seafloor, something is beginning to move. Not the bone itself. The bone is still, settling into the soft sediment that has drifted against it over weeks and months, held in the cold and the dark 2 kilometers below any light. What is moving is invisible. It rises from the bone's surface in a slow, continuous thread. Hydrogen sulfide, a molecule so simple it is made of only 3 atoms, diffusing outward into the surrounding water with the patience of something that has no need to hurry. It spreads in all directions. It climbs. It thins as it goes, its concentration dropping from the dense reactive zone at the bone surface outward to the background chemistry of the open ocean, where the molecule is barely detectable. Between those 2 extremes, along that invisible gradient, the deep sea floor is about to change. This is the moment the sulfophilic stage begins. The word means sulfur-loving. The organisms arriving now are not the large and mobile scavengers that came before, not the dark forms that moved in from kilometers away within hours of the carcass arriving. Those creatures fed and withdrew. They took what was fast and accessible and left. What they left behind was the skeleton, enormous, pale, still holding within its bones a reservoir of stored lipids, so vast that it will take half a century to exhaust. The bones are not empty, they are saturated, and in the cold and the pressure and the total absence of light, the microbes that have colonized that stored fat are beginning to breathe it out as sulfide, and the world around the whale's skeleton is beginning to come alive in a way that has no parallel anywhere else on the deep sea floor. The chemistry begins in the bone's interior. Deep in the cancellous matrix, the porous sponge-like core of a whale's vertebra, or rib, the bone is threaded with thousands of tiny interconnected chambers, each one saturated with pale waxy lipid that accumulated there over the decades of the whale's life. The bone held that fat the way a sponge holds water, not in one place but distributed through its entire interior structure, accessible only from within. Bacteria of the genus Desulfovibrio were among the first to reach it. They are anaerobic, meaning they conduct their metabolism without oxygen, which has long since been exhausted inside the bone's densest chambers. Instead, they draw dissolved sulfate from the surrounding seawater, sulfate that diffuses inward through the bone matrix the way water seeps through stone, and they use that sulfate as an electron acceptor, stripping oxygen away from it in the process of oxidizing the whale's stored lipids. What remains after that reaction is hydrogen sulfide. The bacteria release it the way any organism releases the spent products of its metabolism. It diffuses outward, it finds the water, And there, at the bone surface, the second half of the chemistry begins. Bigiata are the organisms that wait at the boundary. They are filamentous bacteria, long, pale chains of cells that drape across the bone surface in mats so thick they can be measured in centimeters, white as salt, spreading across the whale's ribs and vertebrae, and even onto the surrounding sediment in a living blanket that covers the skeleton entirely. Where disulfovibrio exhale hydrogen sulfide from within the bone, Beggiatoa and the related filamentous sulfur bacteria at the surface oxidize it. They use the energy released by that chemical reaction to fix carbon from the surrounding seawater, building organic molecules from nothing but chemistry and darkness. This is chemosynthesis. It is the same fundamental logic as photosynthesis, but the energy that drives it comes not from sunlight, but from sulfide, from the oxidation of a molecule that would be toxic to most life. The mat does not know this. The mat simply is a dense, white, metabolically active layer conducting primary production in absolute darkness, 2 kilometers below the ocean surface, generating organic matter from chemistry alone, feeding everything above it. The surface of a whale vertebra, in the middle years of the sulfophilic stage, is not bare. It is covered. Every surface is occupied. The Bigiato mat spreads in thick white folds across the pale bone, its texture somewhere between felt and foam, each filament chain only a few cells wide, but the collective mat dense enough to obscure the bone entirely. In some places the mat lifts slightly from the surface, held up by small bubbles of sulfide accumulating beneath it. In other places it lies flat against the curve of a rib, following the bone's contour exactly, as if it has been pressed there. The mat is alive in every part. There is no center and no edge in the metabolic sense. Every filament is conducting the same chemistry, absorbing sulfide from below and releasing it upward as elemental sulfur or sulfate, simultaneously fixing carbon that will flow outward through the food web into every other organism in the community. The mat is the foundation. Without it, the community does not exist. Above the mat and penetrating through it in places, other organisms have taken hold. Among them are the worms that more than any other creature have come to define the whale fall's sulfophilic stage, not because they are the largest or the most numerous, but because they do something that no other known animal does. They have no mouth. They have no gut. They have no anus. Their feeding apparatus is not a feeding apparatus in the ordinary sense at all. It is a root system, a branching, penetrating structure that grows downward into the bone matrix and dissolves the lipids and collagen stored there from within, using symbiotic bacteria held inside specialized cells to conduct the digestion that the worm's own body cannot. From above the bone surface, only the worm's plume is visible. It extends into the water, red or pink or pale, feathery, a few centimeters long, and it pulses gently with the microcurrents of the deep water, absorbing oxygen and exchanging gases with the surrounding seawater. Below the surface, invisible, the root structure spreads through the bone matrix in a slow expanding network, dissolving material that has been stored in that bone since the whale was alive and swimming in sunlit water. These worms are called Osedax. The name means bone devouring.
Osdax were first described in 2004.
A research team from the Monterey Bay Aquarium Research Institute, led by Robert Vrijgenhoek, lowered a remotely operated vehicle to a gray whale carcass resting at 2,900 meters depth in Monterey Bay, and found the bone surface covered with organisms that matched nothing in the existing biological literature. The plumes extended from the bone surface and retracted when disturbed, but when the bone was brought to the surface and examined, The structures penetrating into it were entirely unlike the attachment organs of any known worm. The symbiotic bacteria inside the root cells, responsible for digesting the bone's contents, were heterotrophic. They were not chemosynthetic, did not rely on sulfide, and appeared to consume lipids and collagen directly. The worms had essentially outsourced their digestion to a bacterial community living inside their own tissues, and in exchange had freed themselves from the metabolic constraints that require most animals to maintain a functioning gut. The discovery opened a field of research that has since identified more than 30 distinct Osedax species across the Pacific, Atlantic, and Southern Oceans, each one committed to the same biology, the same rootless underground feeding, the same feathery plume lifting into cold dark water above a bone. It is slowly dissolving from within. The sexual biology of Osedax carries its own quiet strangeness. Each female is the visible organism, the plume, the root system, the gelatinous tube she secretes around herself, anchoring her position on the bone surface. The males are something else entirely. They are microscopic. They never develop beyond a larval form. They live inside the female's gelatinous tube, dozens of them at once, sometimes hundreds, their entire existence reduced to a single biological function. They do not feed. They do not bore into bone. They do not extend plumes into the water or anchor themselves to anything. They live within the tube of a female who is herself anchored to a bone that is itself a remnant of something enormous that once lived in a world of light and air, and within that nested, enclosed existence, they complete their lives. The female's tube is the world they know, and it is sufficient. The biology works. The eggs are fertilized. The larvae disperse into the open water, and in time, some will settle on the next available whalebone and begin the cycle again, the female developing her root system, the males arriving and staying, the gelatinous tube closing around them in the cold and the dark. The sediment immediately surrounding the whale skeleton has changed. In the early weeks, the seafloor near the carcass looked much as it did everywhere else on the abyssal plain, fine-grained, pale gray, nearly featureless, a surface that records only the slowest and most gradual of inputs. Now, in the middle years of the sulfophilic stage, it is darker. The lipids leaching from the bones have saturated the sediment in a spreading halo around the skeleton, changing its color, its texture, its chemistry. Where the outer edge of the halo grades back into ordinary sediment, the transition is gradual, a slow lightening, a thinning of the organic enrichment. But at the center, directly beneath and beside the bones, the sediment is black, with sulfide and dense with microbial activity. Sulfate-reducing bacteria are at work here too, not just inside the bones, but in the sediment itself, metabolizing the lipids that have diffused outward through the pore, water, releasing sulfide into the sediment column, feeding the chemosynthetic system from below as well as within. The chemistry of the bone creates a gradient. This is one of the central organizing facts of the sulfophilic community. Hydrogen sulfide concentration is highest at the bone surface and in the immediately surrounding sediment. It falls as distance from the bone increases, reaching background seawater levels, somewhere between half a meter and a full meter away from the skeleton's edge, depending on the size of the whale and the stage of lipid exhaustion. Different organisms occupy different positions along this gradient according to their tolerance for sulfide and their dependence on it. The Bagiotto occupies the bone surface itself, where sulfide concentrations are highest. Other sulfur-oxidizing bacteria form their own communities slightly further out. Polychaete worms, small crustaceans, and specialized mollusks colonize the sediment within the halo, where sulfide is present but diluted. Further still, organisms less tolerant of sulfide but drawn by the organic enrichment cluster at the gradient's outer edge, feeding on the elevated bacterial productivity without direct exposure to the reactive chemistry at its source. A whale's rib, in this stage, is a different object than it was when the whale was alive. It still has the same form, the long, gentle curve, the dense cortical bone on the outer surface, the porous cancellous interior where the lipids are stored. But its ecological character has been wholly transformed. When the whale was alive, the rib was structural, a support within a body, contributing to breathing and protecting organs and anchoring muscle. Now it is a chemical reactor. It is a slow source of hydrogen sulfide and dissolved organic compounds, a substrate for microbial colonization, a foundation for Osedax root systems and bacterial mats, and the entire food web that depends on them. The rib is still a rib in form. In function, it has become something else entirely, and what it has become will persist for decades until the last of the stored lipid is consumed. The rate of lipid exhaustion is slow, and the stored fat in the cancellous bone of a large baleen whale, a blue whale, a fin whale, one of the great whales that can exceed 20 meters in length, amounts to something between 2,000 and 3,000 kilograms of oil distributed through the skeleton. The sulfate-reducing bacteria that consume it are not fast. They work at the pace of cold chemistry in cold water, their metabolic rates constrained by the temperature, which at bathyal depths rarely exceeds 4 degrees Celsius and can be lower still. The hydrogen sulfide they produce diffuses outward slowly. The Beggiato mat above oxidizes it as fast as it arrives. The balance between production and consumption is the engine of the community, steady and continuous, and it runs for as long as there is lipid to consume. Estimates for how long that takes at a large whale fall range from decades to a century. The skeleton of a whale that died and sank may continue to power a chemosynthetic community in active sulfide production for 50 years or more, in some cases approaching a human lifetime, and in the coldest and deepest waters, potentially beyond it. 50 years is a long time to measure against a single death. The whale that sank was alive, swimming, breathing, feeding at the surface in some part of the open ocean, and its body fell and settled and began this long, slow process of transformation. By the time the last of the lipid has been consumed from its bones, the world above the ocean's surface will have changed in ways that bear no relation to anything present at the moment of its death. The sulfophilic community is patient in a way that has no human equivalent. Not patient because it waits, but patient because it exists on a time scale where the distinction between fast and slow has dissolved. The bacteria do not know they have been working for decades. The Osedax worms boring through the bones are not aware that the structure they inhabit is centuries older than the ecosystem it now supports. Time at the whale fall is simply the medium in which chemistry occurs. Within the whale bones themselves, in the deepest and most airless chambers of the cancellous matrix, there is a second chemistry running alongside the sulfide system. Archaea, a domain of life distinct from bacteria, neither prokaryote nor eukaryote in the ordinary sense, have colonized the most anaerobic zones of the bone interior and are conducting methanogenesis. They consume hydrogen and carbon dioxide and produce methane, the same gas that forms in swamps and bogs and in the guts of large animals, released as a metabolic byproduct of their anaerobic respiration. Some of the methane diffuses outward through the bone and into the sediment and more methanotrophic bacteria. Bacteria that oxidize methane as their energy source can consume it in a process that mirrors the sulfide oxidation above, generating a second chemosynthetic energy input to the community. Whether the methane system at most whale falls is large enough to support a significant independent food web is not fully resolved, but the architecture is there. The chemistry is running. In some whale falls, and possibly in others not yet examined with the right instruments, the methane pathway may extend the productive life of the community beyond what the sulfite system alone could sustain, adding another few years or another decade to the long, slow output of chemical energy from a single whale's bones. The water above the whale skeleton in the middle years of the sulfophilic stage is not empty. It holds the community in suspension, both literally and figuratively. Small crustaceans move through the water column just above the bone surface, feeding on the bacterial mat and on each other. Polycate worms extend tentacles or feathery feeding structures into the current, harvesting particles of organic matter that drift upward from the mat below. Gastropod mollusks move slowly across the bone surfaces, grazing on the microbial film in the spaces between Osedax tubes. The community has a structure in 3 dimensions: the mat at the base, the sediment community spreading outward in the sulfide halo, the water column community occupying the space above, and in the upper reaches of that column, the transition zone where the chemistry of the whale fall gives way to the chemistry of the surrounding deep ocean, and organisms from both worlds can be found together at the boundary. A single Osedax female, anchored to the surface of a whale vertebra, has been in place for weeks or months. Her gelatinous tube has extended slightly with the slow growth of her root system below the bone surface. Her red plume extends above the tube, 3 or 4 centimeters into the water, its fine filaments absorbing oxygen from the current. Inside the tube, the males she carries have fertilized recent batches of eggs, and the larvae from those eggs have dispersed into the open water above the seafloor, carried away on whatever faint current moves through this part of the abyss. Some will settle nearby, on other bones of the same skeleton. Some will drift further and settle nowhere useful, expending their short larval energy in open water above soft sediment where no bone exists. A small number will find another whale fall, kilometers away, and anchor there, and begin the same process again, penetrating bone and hosting symbiotic bacteria and secreting a gelatinous tube around a community of microscopic males who will live out their existence in that enclosed world. The Begiata mat, seen from above, from the perspective of a remotely operated vehicle descending toward the skeleton on a research expedition, is white against the darkness. The bones themselves are pale, but the mat is whiter still, a thick, slightly irregular blanket draped across the entire skeletal structure, following its contours, pooling in the low points between ribs, thinning where the current runs faster across the exposed surfaces, accumulating where the flow slows, and sulfide can concentrate. The mat is not uniform in its whiteness. Where sulfide supply is highest, the mat is thickest and brightest, almost luminous in the AeroV's lights. Where the mat thins toward the edges of the bone surface, it transitions through gray and pale brown into the bare sediment beyond, and the transition carries the feeling of a shoreline, a boundary between 2 worlds, the chemical world of the whale fall and the biological desert of the surrounding abyssal plain, meeting in a gradual fade. The soft sediment 5 meters from the nearest whale bone is for most purposes the same as it was before the whale arrived. The background marine snow, the slow, sparse rain of organic particles drifting down from the surface, settles there as it always has, at rates too thin and dilute to support much life. A polycate worm may burrow through the top few centimeters. A foraminifera may extend its pseudopods into the sediment in search of a particle of organic matter. The biological productivity is low. The oxygen in the sediment is present throughout. The chemistry is the quiet chemistry of the ordinary abyss. 5 meters away, just inside the influence of the whale fall's lipid halo, the sediment is transformed. The difference is not gradual so much as it is a slow brightening. One can move outward from the skeleton and feel the community thinning, the chemical signature fading, and then pass a threshold where the whale fall's influence simply does not reach, and the ordinary seafloor resumes, as quiet and sparse as it was before the whale arrived. The hydrogen sulfide diffusing outward from the bone is toxic to most aerobic life, and the community of the sulfophilic stage is composed almost entirely of organisms that have evolved specific mechanisms to tolerate or exploit it. The Beggiatoa carry sulfide-binding proteins. The polychaete worms in the sediment halo maintain specialized hemoglobins that bind sulfide without allowing it to interfere with oxygen transport. The Osedax worms exposed to sulfide at the bone surface manage their exposure through their root structures, limiting the direct contact between their tissues and the reactive chemistry below. The community is not tolerant of sulfide in the way that some organisms are tolerant of cold or of depth, tolerant in the general sense of being hardy, adaptable. These organisms are tolerant of sulfide specifically through specific evolved biochemistry, because the ancestors of each of them spent generations at whale falls and at hydrothermal vents and at cold seeps, the other places where sulfide is produced in the deep ocean, and only those lineages that could manage the chemistry survived long enough to reproduce in those places. Some of the species present at a whale fall in the sulfophilic stage are the same species found at hydrothermal vents, those places on the seafloor where the heat of the planet's interior drives superheated water and minerals through cracks in the crust. The vents are distant from any whale fall, separated by hundreds or thousands of kilometers of open, featureless abyssal plain, where the seafloor provides no chemical energy source. Between a vent field and the nearest whale fall, a larva drifting in the deep water current would need to travel far and settle somewhere useful. The whale falls distributed across the deep ocean floor may function as stepping stones, intermediate habitats where chemosynthetic species can persist and reproduce between one vent field and another, maintaining genetic connection across distances that would otherwise isolate populations. The whale fall, in this view, is not just a local event. It is part of the architecture of the deep ocean's biological connectivity, a link in a chain that runs across the basin floor over geological time. The bones of the whale that is now a whale fall were once growing. They were laid down over the whale's long life. Year by year, the cancellous matrix filling with lipid as the whale fed and fattened, the dense cortical outer bone thickening and strengthening with use. The whale carried those bones through cold and warm water, through thousands of miles of open ocean, through seasons and feeding grounds and migrations. The lipids stored in the bone matrix accumulated from every krill swarm and fish school the whale ever consumed, converted and deposited and held in the bone the way a tree holds carbon in its wood. The bone was a storage system, passive and internal, unnoticed by the whale that carried it. Now 2 kilometers below the ocean surface, in the dark and the cold, those stored reserves are being consumed by the organisms that have colonized the bones. The energy that passed into the whale from the ocean's surface, from sunlight and phytoplankton, and the upward flow of nutrients from the deep, is passing out of the whale's bones and into the bodies of bacteria and worms and crustaceans on the seafloor, and from them outward into the wider community. And from there, eventually back into the dissolved organic carbon of the deep ocean water, dispersing into a medium that will carry it to other places and other organisms over the long, slow circulation of the deep ocean. Nothing is lost. The energy changes form. The carbon in the whale's bone lipid becomes the carbon in a Beggiata filament, and then the carbon in the body of a polychaetary worm that grazes on the mat, and then the carbon in whatever organism consumes that worm, and so on along the food web until the carbon has been metabolized and released into the water as carbon dioxide and rejoins the dissolved inorganic carbon pool of the deep ocean. Some small fraction may eventually reach the surface through deep water upwelling, where it can be fixed again by phytoplankton in a sunlit surface layer somewhere far from where it began. The whale's life fed the ocean's surface once. Through the nutrients, its excretions return to the water. Its death feeds the ocean's depth for decades through the slow chemical release of stored energy from its bones. The Osedax worm boring through a whale vertebra is dissolving bone that was laid down in a living whale decades ago. The lipid it is consuming was stored in that bone over the course of that whale's adult life. The symbiotic bacteria inside the worm's root cells are consuming collagen that held the bone's mineral matrix together from the inside, collagen that was synthesized by the whale's own osteoblasts in the years when the bone was still growing and being maintained by a living body. The worm does not know any of this. The worm simply grows its root system through the porous matrix, following the lipid concentration gradient deeper into the bone where the richest reserves are held. But the continuity is there regardless. What the whale was in life is what the worm eats in death, and what the worm is in its turn feeds the larvae that disperse from the whale fall and carry the genetic memory of Osedax biology out into the open water of the deep ocean to settle eventually on the next available bone in the next available darkness. The whale fall, in the middle of the sulfophilic stage, is a place of extraordinary density, in a world of extraordinary sparseness. The abyssal plain surrounding it receives, on average, only a tiny fraction of the organic production that falls from the surface. Most of it is remineralized in the water column before it reaches 2 kilometers depth, and what arrives at the seafloor is diluted across vast areas of featureless soft sediment. The whale fall concentrates what took the whale its entire lifetime to accumulate, hundreds of thousands of kilograms of soft tissue and bone lipid, into a single point on the seafloor, and releases it slowly over 50 years or more. The contrast with the surrounding seafloor is immense. The whale fall community may contain hundreds of species and millions of individual organisms in a space that could be crossed in a few minutes' walk, surrounded by a seafloor that might otherwise support only a handful of species and a fraction of that biomass across the same area. The whale fall is a reef in the desert, a brief and localized abundance in a world where abundance is the exception. The long middle of the sulfophilic stage has no obvious climax, no turning point, no event that marks its center. The chemistry continues. The mat grows and thins and grows again as sulfide supply fluctuates with the rate of lipid consumption. The Osedax worms bore deeper into the bones as the surface layers are exhausted, following the richest lipid inward and downward. The sediment halo expands slowly outward as lipids continue to leach from the bones into the surrounding pore water. The community changes composition gradually as different species colonize at different times, some arriving early, some establishing only after the mat has reached a certain density, some present only in the middle years when sulfide production is at its peak. There is no steady state, exactly. There is a long, slow arc, a building and a peak and a gradual decline measured not in seasons but in decades, the tempo set by the rate at which cold bacteria can consume cold wax in cold darkness. And then, slowly, the sulfide production begins to thin. The richest lipid reserves, the ones closest to the bone surface and to the Osidax roots, have been consumed first. What remains is deeper in the bone matrix, less accessible, requiring the root systems to extend further to reach it. The Beggiato amat, now receiving less sulfide than it did in the peak years, thins gradually, the white blanket becoming patchy, the mat retreating from the outer surfaces of the bones first, then from the lower surfaces, concentrating finally over the areas where sulfide production continues longest. The polyseed worm density in the sediment halo declines as the lipid enrichment of the sediment fades. The small crustaceans that grazed on the mat find less to consume and disperse or die. The community does not collapse at once. It fades. It thins and quiets over years, and then over more years, the chemical energy source declining at the same slow pace at which it was first established. What remains when the sulfophilic stage has run its full course is still a skeleton. The bones are thinner than they were, hollowed further by the Osidax root systems, their cancellous matrix partially dissolved, the cortical outer surface pitted and roughened by decades of microbial colonization. But the form persists. The curve of the ribs is still there. The mass of the vertebrae still traces the line of the whale's spine along the seafloor. The bones are lighter, more porous, more fragile, but they remain a structure recognizable as what it was, anchored in the soft sediment of the abyssal plain, surrounded now by sediment that has returned gradually to the quiet chemistry of the ordinary deep ocean as the last of the lipid enrichment dissipates. The Osidax worms are still boring. They will continue for a while, working the deepest and least accessible lipid reserves, their root systems reaching into crevices and channels that have not yet been exhausted. The mat has gone thin and patchy. The sediment halo has faded to ordinary gray. The community that was there, the hundreds of species, the millions of individuals, the decades of chemosynthetic primary production in complete darkness, has dispersed or died, its members leaving no permanent record on the bone except the pitting and dissolution that marks where they fed. The whale's bones, in the long stillness that follows, hold within them the faint chemical ghost of what they once were. The lipid is nearly gone. The collagen is partly consumed. The mineral matrix remains, calcium and phosphate, the same chemistry that built the bone inside a living whale. In time, more time still, years beyond the years already spent, the bones will become substrate. Their surfaces will be colonized by encrusting organisms, looking not for chemical energy, but for hard ground in a soft sediment world. And the bones will become part of the benthic community in a different way, hosting suspension feeders and encrusting invertebrates and the quiet life of a permanent hard-bottom site. But that transformation is still in the future. For now, the whale's bones rest on the seafloor in the deep quiet, the last of the Osidax plumes extending above the bone surface in the faint current, red and slow, absorbing oxygen from water that has not seen the sun in 2 years. The whale that grew those bones did so in a world of light and air and motion. It breathed and fed and traveled and died in the open ocean, and its body fell through 2 kilometers of dark water and came to rest here, and for 50 years it sustained a community that the sunlit world above will never see and cannot easily imagine. The Osidax worm dissolving the last of the collagen from a vertebra that once helped a living whale breathe is completing a process that began before it was born and will continue in the form of dispersing larvae and settling spores and the slow turnover of carbon in the deep ocean, long after the bone itself has been fully incorporated into the sediment. The whale's bones are old and nearly spent and still feeding something. They will feed something until there is nothing left to feed, and then the sediment will close over them, the way the deep ocean closes over everything, slowly, completely, without haste. In the Santa Cruz Basin, on a stretch of seafloor that receives almost no light and almost no visitors, a single site was cataloged in the year 2002. The researchers who descended to it counted the animals living there, named the ones that could be named, and set aside the ones that could not yet be placed. When the count was finished, more than 200 species had been found at one location on an otherwise nearly empty plain of sediment. Some of those species had never been recorded anywhere else on the ocean floor. Some had no name. Some were so small and so still that they seemed to belong less to the world of animals than to the world of accumulation, of slow gathering, of matter that had found a way to organize itself in the dark. 200 species at a single site. That number settles differently when held quietly than when spoken quickly. On the abyssal plain surrounding that spot, a square meter of seafloor might support a handful of organisms, sparse, patient creatures adapted to living on the slow drift of particles from the surface, 2 kilometers above, a diet so dilute that metabolisms must be stretched across long time to make use of it. And then, within the boundaries of one whale's body, this, a density of life that has no ordinary explanation on the deep sea floor. An exception that is, in its own way, a small world. The whale that made that world had been gone for years before the researchers arrived. Only its bones remained, and the animals that had settled onto and into those bones, and the chemistry still moving slowly outward from the lipid-rich cancellous matrix deep inside the skeleton. The whale's name, in life, would have been a species name: gray whale, blue whale, fin whale, something that could be said in 2 words, but on the seafloor it had become something that takes longer to describe. It had become a place. A place takes time to make. What was found at that Santa Cruz Basin site in 2002 had been building for years before anyone came to count it. The soft tissue had been gone for longer than that, stripped in the early weeks and months by large scavengers converging from the surrounding dark. The enrichment opportunists had come after, colonizing the exposed bone surfaces in dense mats. And then, beginning slowly and persisting for decades, the sulfophilic community had taken hold, its chemistry powered by the slow bacterial reduction of sulfate in the lipid-soaked bone matrix, the hydrogen sulfide produced by that process diffusing outward through the sediment in invisible gradients that different organisms occupied according to their precise tolerances. By the time the researchers counted 200 species, the whale had been feeding the deep sea floor for what was probably 10 or 20 years. It would continue for decades more. Within a few years of that survey, a description was published of a worm found not in the Pacific, but in the North Atlantic, on the seafloor of a shallow Swedish fjord. Researchers had placed the bones of a minke whale on the bottom of Kosterfjord at a depth of roughly 400 feet, and waited. What colonized those bones was a new species of bone-eating worm, kin to the ones being described in Monterey Bay at almost exactly the same time. The new species was given the name Osidax mucofloris, meaning mucus flower, for the feathery translucent plumes it extended into the water above the bone surface, while its root-like structures bored silently downward into the matrix below. It fed on what was locked inside the bone: lipids, collagen, the stored chemistry of a whale that had lived and died in northern waters. and it did so in the same way its Pacific cousins did, through symbiotic bacteria that carried the actual metabolic work living inside the root tissues of a worm that had no mouth, no gut, and no need for either. The Atlantic and Pacific species were separated by ocean and by the long cold record of their separate evolutionary histories. The biology, at its core, was the same bone-eating strategy, the same root-like penetration, the same radical sexual dimorphism, microscopic males living in dozens inside the gelatinous tube of a single female, their entire existence reduced to one quiet function. The same answer, found twice, to the same opportunity, a whalebone on a seafloor, saturated with decades of slow chemical fuel. Life, given enough time, finds a way to this. It does not try to find it. It simply arrives because the lineages that arrived persisted, and the ones that could not use what a whale's bones contain did not persist at the whale's bones. And over time, the community that gathers at a whale fall is a community shaped by this exact pressure, this exact chemistry, this exact depth, and cold and dark. The genus Asterisk, Osidax asterisk, now contains more than 30 described species. They have been found in the Pacific, the Atlantic, and the Southern Ocean. They have been found at depths of nearly 10,000 feet, and at depths shallow enough that some light still reaches the seafloor. Every one of them converges on the same ecological role: bone eater, lipid miner, root driller, mucus flower. They are a lineage that diversified across the ocean floor, the way that other lineages diversify across mountain ranges or river systems along the gradient of what is available, what is cold enough, what is deep enough, what holds enough stored energy to sustain a sessile animal through the long years of the sulfophilic stage. The question of how they spread, how any single population reaches a new whale fall when whale falls are separated by miles of open sediment, is answered by larvae. Tiny, drifting, broadcast into the water column by spawning adults, these larvae travel on the slow currents of the deep ocean and settle where the chemical signals draw them. The diffuse plume of sulfide and dissolved lipid products spreading outward from a whale skeleton in the dark. If the larvae travel far enough, and if a new whale carcass lies within their range when they are ready to settle, the population extends. A network of whale falls, spaced close enough that larvae can move between them, is a network of connected habitat islands, not in the biological sense of islands separated by water, but in the ecological sense of suitable patches separated by unsuitable ground. On the pre-whaling North Pacific floor, the best estimates place whale carcasses at intervals of between 5 and 16 kilometers in the most productive regions of the basin, 5 to 16 kilometers. Within that range, the larvae of many deep-sea invertebrates can drift on current and settle. The network at those spacings was dense enough to be continuous in a biological sense, each whale fall reachable from its neighbors, each one a stepping stone. Populations could flow, genes could move. A species that evolved at a vent field could colonize a whale fall, and then another whale fall, and then another, spreading across the basin without ever crossing the barren sediment plain directly, moving instead through a series of temporary oases that formed and faded across decades. This is the stepping stone hypothesis, and it carries an implication that takes a moment to settle. If whale falls are stepping stones for the chemosynthetic fauna of the deep sea, then the number of whales matters, not just as a count of living animals, but as a count of future seafloor ecosystems. Each living whale carries within its bones the substrate of a community that will take decades to fully assemble, and may support species found nowhere else in the surrounding ocean. Each whale that completes its natural lifespan and sinks into the deep adds one more stone to the network, one more node in the web of habitat that chemosynthetic species depend on for dispersal. Commercial whaling reduced blue whale populations to somewhere between 1 and 3% of their numbers before the industry began. The open ocean, once crossed by tens of thousands of blue whales, was reduced to a remnant. The annual input of whale biomass to the deep sea floor, the total mass of whale carcasses settling across the world's ocean basins in a given year, fell by a factor that may be as large as 2 or 3. The stepping stone network thinned. In some regions, the spacing between whale falls would have grown from the 5 to 16 kilometers that allows larval movement to distances far too large for most larvae to cross. Nodes in the network went dark, not through any visible catastrophe on the seafloor, but through the simple arithmetic of fewer whales dying and fewer carcasses sinking. What that meant for the species that depended on those stepping stones is not fully known. The deep sea floor is vast and difficult to study, and the baseline community of a pre-whaling abyssal plain is not something any researcher has ever observed directly. The record that would show what was lost, what species spread through the North Pacific on a network of closely spaced whale falls, and then contracted or vanished when that network thinned, is mostly absent. The seafloor does not leave the kind of archive that makes a before and after comparison easy. What remains is inference, the genetic structure of living populations, the distribution of species across known whale fall sites, the dispersal ranges of larvae measured in laboratory conditions and checked against population patterns in the field. From those fragments, a picture emerges. Bathymedialid mussels, the same genus found clustered at hydrothermal vents in the Pacific and the Atlantic, have been collected at whale fall sites, living through the sulfophilic stage on the chemistry of hydrogen sulfide diffusing from whale bones. Their population genetics carry a signal. The pattern of genetic variation within and between populations is consistent with stepping stone dispersal. Individuals moving from whale fall to whale fall across a network of sites rather than long-distance direct transport between distant vent fields. The mussels, in other words, did not simply drift from one vent to the next across hundreds of miles of open ocean floor. They moved through intermediate sites. They used the whale falls, and the genetic record, read carefully, tells the same story as the larval biology and the geographic spacing of known sites. Whale falls are functional links in the dispersal map of chemosynthetic life, not incidental additions to it. The connection between whale falls and hydrothermal vents is one of the more quietly compelling structural facts of deep-sea biology. Vents are permanent, or nearly so. They persist for decades to centuries at individual locations, powered by the slow exchange of heat between the ocean and the planet's interior. Whale falls are temporary, lasting decades, and then fading as the lipid reserves are exhausted and the chemosynthetic community contracts. But in terms of the chemistry they support and the community they host, the two systems are closely related. Both sustain primary production through the oxidation of reduced sulfur compounds. Both are dominated by organisms that evolved the ability to live without sunlight, using the energy in chemical bonds instead of the energy in photons. Both host communities of mussels, clams, polychaety worms, and microbial mats that belong to lineages found at the other system as well. The relationship between the two systems is not simply ecological overlap, it may be evolutionary connection. The hydrothermal vent fauna of the deep Pacific is old. Some lineages date back tens of millions of years, to a time when the whale was already an ocean animal, but not yet the massive, deep-diving creature it would become. The ancestors of the bone-eating worms and the sulfide-adapted mussels that now colonize whale falls may have moved between vent systems across the ancient ocean floor, using whatever stepping stones the Eocene and Oligocene oceans provided, cold seeps perhaps, or the bones of smaller marine mammals, or organic matter from the surface, on a scale and schedule that cannot be reconstructed from the present. What is clear is that the chemosynthetic fauna of the deep ocean is a lineage with deep roots, and that whale falls, when they became abundant enough to use, were incorporated into the dispersal geography of that lineage in a way that left traces in the genetics of living populations. A vesicomiid clam, resting at the base of a whale vertebra in the sulfophilic stage, carries within its tissue the same symbiotic sulfur oxidizing bacteria that its relatives carry in vent fields thousands of kilometers away. The clam is not from the vent. It did not travel there directly. Its population traces through whale falls and cold seeps and whatever other reduced sulfur habitats were available to it along the route, each generation dispersing as larvae, each larva settling where the chemistry was right. The cumulated geography of 10,000 generations, written in the slow divergence of its genome from the genome of its vent-dwelling cousin. It rests on the bone of a whale, drawing sulfide from the bacteria reducing lipids in the matrix below, and it belongs equally to the ecology of the whale fall and to the ecology of the deep ocean as a whole, a creature that exists at the intersection of the temporary and the permanent, the specific death of one animal and the ancient persistence of an entire chemosynthetic world. The whale fall at the center of all of this, the actual skeleton, resting on the seafloor, changes over the decades it persists. The sulfophilic stage does not end all at once. The lipids in the cancellous bone matrix are not consumed uniformly. Different bones, the vertebrae, the ribs, the skull, have different lipid concentrations, different densities, different rates of bacterial colonization and chemical depletion. Some bones may sustain active chemosynthesis for 20 years after others have been largely spent. The sulfophilic community contracts gradually across the skeleton as the fuel is drawn down, contracting toward the richest bones, persisting longest in the densest parts of the cancellous matrix. The mats of filamentous bacteria thin. The populations of bone-eating worms decline as the collagen and lipid they depend on are exhausted. The hydrogen sulfide gradient fades. What remains after the sulfide chemistry ends is the skeleton itself. Cleaned and largely mineralized, the bones of a large whale are a substantial architectural structure on an otherwise nearly featureless seafloor. They are hard substrate in a world of soft sediment, and hard substrate in the deep sea is rare. The organisms that require it, the suspension feeders, the encrusting invertebrates, the corals and sponges and feather stars that anchor to solid surfaces and extend their feeding structures into the water column, find few places to settle on the flat abyssal plain. A rocky outcrop created by geological activity is one, a whale skeleton is another. In the final stage of the whale fall, the bones are colonized by these suspension feeders, their surfaces slowly encrusted with pale corals, their angles furred with the fine branches of sponge colonies. The spaces between ribs occupied by communities of encrusting invertebrates that would be at home on a rocky reef. The skeleton stops being a whale fall in the ecological sense, no longer a discrete temporary ecosystem, no longer defined by the chemistry of decomposing lipids, and becomes instead a permanent piece of hard substrate integrated into the surrounding benthic community. A reef of a kind. A rocky outcrop built not from basalt or limestone, but from the cancellous bone of an animal that once breathed air at the surface of the same ocean directly above. From the surface, looking down, there is nothing to indicate that any of this is happening. The ocean is featureless from above, a vast plain of water with no visible seam, no visible depth, no visible sign of the communities living on its floor. A ship passing over the Santa Cruz Basin site, cataloged in 2002, would see open water. Sunlight would reach only the uppermost few hundred feet of the water column before being absorbed. Below that, darkness. Below that, more darkness, and then more, and then the slow descent into the bathyal zone where the temperature steadies near freezing, where the pressure is measured in hundreds of atmospheres, where the light has been absent for so long that there is no word for the kind of absence it represents. Not night, not shadow, not the darkness of a room with the curtains drawn, but a darkness that has never in the history of that water been broken by a photon from the sun. In that darkness, the whale falls. In that darkness, the community assembles. In that darkness, the chemistry runs for decades on the fuel of stored lipids. The hydrogen sulfide diffuses outward in gradients too subtle to feel. The larvae drift and settle and metamorphose into the bone-eating worms and sulfide-adapted mussels and filamentous bacteria that make a temporary world out of one animal's death. None of it is visible from the surface. None of it requires the surface to take notice. It proceeds with the patient indifference of chemistry and biology operating on their own timescales, in their own register, in a part of the planet where the concept of day has never applied. A large whale swimming at the surface today, a blue whale, moving slowly through cold, productive water, its lung capacity enough to take a breath that will sustain it through a long dive, its baleen plates filtering tons of krill across a feeding season, carries within its skeleton the substrate of a future ecosystem. This is not a metaphor. The cancellous bone matrix of a blue whale contains between 2,000 and 3,000 kilograms of pale wax-like lipid stored there during the whale's life and accessible to the bacteria that will colonize it after death. That oil is already there, already present, already sufficient to sustain decades of chemosynthetic production on whatever seafloor the whale eventually reaches. The ecosystem is latent. It waits. It does not wait in any purposeful sense. The whale is not carrying its future whalefall towards some intended site. The bacteria that will one day colonize its bones are not waiting specifically for it, not orienting toward it, not aware of it in any sense that the word awareness can accommodate. But the chemistry is already in place. The fuel is already stored. The network of chemosynthetic organisms spread across the deep ocean floor includes populations that have evolved over millions of years to detect the chemical signals of a whale carcass settling in the dark. The diffuse plume of dissolved amino acids and lipid compounds spreading outward through cold water, and to move toward it, and to settle, and to begin the slow process of disassembling a life into the materials for other lives. There is something in this that resists the ordinary categories of beginning and end. A whale is born into a world of sunlight and air, grows across decades, feeds in cold, productive waters, breathes at the surface in long, slow exhalations that can be heard from a considerable distance on a calm day. It lives in the lit part of the ocean, and then at the end of its life, it becomes the foundation of a community in the dark part, a community more diverse than most of the surrounding deep-sea floor, a community that will persist for longer than most human institutions, a community that may serve as a stepping stone for species whose dispersal geography spans the whole Pacific Basin. The whale's life and the whale's death are not separate events. They are one extended process, the surface part and the deep sea part, continuous with each other across the boundary of the sea floor, in the same way that a river is continuous across the boundary where it enters the ocean. The deep sea floor is not a barren place that occasionally receives extraordinary inputs. It is a place organized in part around those extraordinary inputs: the whale falls, the cold seeps, the hydrothermal vents, and the long intervals of sparse marine snow drifting down from above that sustain it between them. The whale falls are not exceptions to the ecology of the abyss. They are part of the structure of it. They are nodes in a network that has been operating at various densities and spacings for as long as whales have been dying in the deep ocean and sinking to its floor for tens of millions of years across successive whale lineages, through the slow drift of continents and the opening and closing of ocean basins. In the pre-whaling ocean, a whale swimming in the North Pacific might eventually become a whale, fall within 5 to 16 kilometers of another whale fall. The larva that hatched from an egg laid on one skeleton might drift to the next, settle, and in turn produce larvae that reached the one beyond. The network had the density of a road system, not a direct route between any 2 points, but a connected infrastructure, a way of moving across the floor of the ocean that did not require crossing the open sediment plain in a single generation. Species spread, populations mixed, genetic diversity moved laterally across the basin, carried by the slow drift of larvae between temporary oases of concentrated chemistry. That network is thinner now. The whales that would have died naturally and sunk to become whale falls are largely absent from the regions where they once moved in large numbers. The annual input of whale biomass to the deep sea floor has declined substantially from what it was before the industrial harvest of the 19th and 20th centuries. The stepping stones are more widely spaced. Some connections in the network that once existed may no longer function, the gap between suitable sites now too large for most larvae to cross in a single generation. The consequences of this thinning are not fully visible in the data that exists, because the deep sea floor is difficult to survey, and because the baseline, the ecology of the pre-whaling deep ocean, was never directly studied. It can only be inferred from the traces that remain. What can be said with some confidence is this: the deep sea floor of the North Pacific supported a more connected chemosynthetic community when large baleen whales were abundant than it does now. The dispersal routes that some species relied on were partly made of whale falls. Where those falls are absent, the routes are interrupted. Whether the interruption has caused local extinctions, reductions in genetic diversity, or simply slower dispersal across the basin is a question that the available evidence cannot yet fully answer. It is a question that researchers are still assembling the tools to ask properly. The whale falls that do exist, the ones created by whales dying of natural causes, the ones deliberately created by researchers who sink whale carcasses in monitored locations, the ones accidentally created when stranded animals are towed offshore and released in deep water, each create a local surge of diversity on an otherwise sparse seafloor. Each one supports its community for decades. Each one may serve as a node in the dispersal network for some fraction of the chemosynthetic species that find it. And each one, when the lipids are finally exhausted and the sulfide chemistry fades and the bones are encrusted with corals and sponges and integrated into the surrounding benthic community, leaves behind a small alteration of the seafloor that persists long after the whale fall ecology has ended. A piece of hard substrate on a soft sediment plain, a slight enrichment of the local sediment chemistry from decades of lipid leaching, a small raised feature that breaks the otherwise flat topography of the abyssal floor. These are the permanent signatures of a whale fall, not the dramatic ones, not the dense living community of the sulfophilic stage, but the quiet structural changes that remain after everything else is gone. A reef that was once a whale, a slightly enriched sediment patch that was once the margin of a chemosynthetic ecosystem, a place where the deep sea floor is in some small way different from what it would have been had no whale ever settled there. On the floor of the Santa Cruz Basin, in the darkness and the cold and the pressure, the site cataloged in 2002 is still there. The specific whale that made it has long since been dissolved back into the chemistry of the ocean, its lipids consumed, its collagen metabolized, its bone mineral slowly reintegrating into the surrounding sediment over the long timescale of geological process. The more than 200 species found there have moved on in various ways. Some have died, some have dispersed as larvae to other sites. Some may still be present in reduced numbers on the enriched sediment that remains. The bones, encrusted now with suspension feeders, are a small reef in the dark. Nothing marks the place from above. Nothing at the surface indicates what has been happening on the floor below. But something happened there. For decades, in a part of the ocean where most of what occurs is slow and sparse and dilute, one site sustained a dense and diverse living community powered by the chemistry of a single animal's death. More concentrated carbon arrived at that spot in a single settling event than the surrounding seafloor accumulates from marine snow across centuries. More species lived within the boundaries of that one carcass than in a much larger area of the surrounding abyssal plain. In more ecological events, predation, competition, reproduction, colonization, succession occurred at that location in 50 years than would have occurred in the surrounding sediment across a far longer span. The scale of what one death can sustain is one of the quieter truths of deep-sea biology. Not quiet because it is small, but quiet because it happens in a place where quiet is the only register available. The deep ocean does not amplify, it does not broadcast, it does not announce the 200 species on the sea floor of the Santa Cruz Basin, or the genetic record of dispersal written in the mussels resting at the base of whale vertebrae, or the slow conversion of a blue whale's stored lipids into decades of chemosynthetic production in complete darkness. These things occur without any surface visible to them, without any light to illuminate them, without any witness except the organisms carrying them out, and the occasional, brief, illuminated visit of a remotely operated vehicle descending on a tether from a ship above. Every large whale swimming in the ocean now carries within its cancellous bone the latent substrate of a future whale fall ecosystem. The lipids are already stored. The potential community is already distributed across the deep sea floor, its members living at existing whale falls and vent fields and cold seeps, producing larvae each season that drift on deep currents and settle where the chemistry invites them. The connection between a whale at the surface and a whale fall on the sea floor is not interrupted by the death, it is completed by it. The whale's life feeds the sunlit ocean. The whale's death feeds the dark one. Both are the same animal, the same stored chemistry, the same accumulated decades of feeding and growing, moving from one part of the ocean to another across the boundary of the seafloor. What arrives at the bottom in the slow billow of disturbed sediment that marks the end of the descent is not simply a carcass, it is a future. A temporary but dense future, lasting 50 years or 100, populated by hundreds of species, driven by chemistry that will produce hydrogen sulfide from stored lipids for decades in the absolute dark. It is one of the most concentrated expressions of biological possibility in the deep ocean, a discrete site where life for a bounded time runs at a density and diversity that the surrounding floor cannot match and may not approach again for a very long time. And then it fades. The lipids are consumed, the sulfide gradient dissipates, the microbial mats thin, the bone-eating worms, finding less and less collagen in the increasingly mineralized matrix, produce fewer larvae, sustain fewer adults, retreat from the bones they have spent decades drilling. The mussels, no longer bathed in the sulfide concentrations their symbiotic bacteria need, find the site less hospitable. The mobile scavengers of the early weeks are long gone. The enrichment opportunists of the middle period have dispersed or died. What remains is a skeleton encrusted with the permanent patient suspension feeders, the corals growing slowly in the cold dark, 1 millimeter of calyx per year, building structure that will outlast by many times the whale that made the site possible. The whale is gone, in every meaningful chemical sense, long before the corals stop growing, but the corals continue, the sponges continue.